Hey guys, tonight we're going somewhere you've never been before. Somewhere dark, acidic, and absolutely swarming with life. We're diving deep into your own body, specifically into the chaotic, microscopic battleground of your gut. And here's the twist. You're not you anymore. You're a single bacterium, fragile, outnumbered, and fighting for survival in one of the most hostile environments on Earth. Forget the castles and empires we've explored before. Tonight's kingdom is your intestines, and trust me, you wouldn't last an hour. So, before you settle in, take a moment to like the video and subscribe, but only if you genuinely enjoy what we do here. And let me know in the comments where you're tuning in from and what time it is for you. It's always fascinating to see who's drifting off to sleep alongside these stories from around the world. Now, dim the lights, maybe grab some water, ironically, and let's shrink down together into the warm, wet, utterly unforgiving world inside you. Congratulations. You've just been born as a bacterium in the human gut. The good news? You're alive. The bad news? Everything around you is trying to kill you. Right now, inside your body, there are roughly 39 trillion bacterial cells. That's more bacteria than human cells. They outnumber you in your own skin. And every single one of them is locked in a constant struggle for space, food, and survival. You might think your gut is a peaceful place, a quiet tube where food gets digested and nutrients get absorbed. But if you were the size of a bacterium, you'd see the truth. It's a war zone, a churning, acidic, immune-patrolled hellscape where death comes from every direction. Chemical weapons from your liver, assassins from your immune system, competitors who've lived here for years and aren't interested in sharing. And yet, somehow, trillions of bacteria survive. They've adapted to thrive in conditions that would melt most life forms on contact. They've built cities, formed alliances, and developed weapons we're only beginning to understand. But survival isn't guaranteed, not even close. So let's see if you'd make it. Let's walk through a day, maybe an hour, in the life of a gut bacterium. And let's find out why your body's most populated neighborhood is also its most dangerous. Welcome to the gut. Try not to dissolve. You didn't choose to be here. You were swallowed. Maybe you hitched a ride on a piece of lettuce that wasn't washed thoroughly enough. Or you were living peacefully on someone's unwashed hand before they touched their mouth. However you got here, the moment you crossed those lips, your fate was sealed. Welcome to the human digestive system. Try not to panic. The first thing you notice is the temperature. It's warm, roughly 37 degrees Celsius, which is about 98.6 Fahrenheit. For a bacterium, that's actually comfortable. Your enzymes work well at this temperature. Your cell membrane stays fluid. You might even think you've lucked out, but then the chewing starts. Teeth the size of buildings crash down around you. If you're on that lettuce leaf, you're being pulverized by molars that can exert a force of around 70 pounds per square inch. The world becomes chaos, crushing, grinding, tearing. Most of the bacteria that entered with you are already dead, their cell walls ruptured, their contents spilling into the saliva that's now flooding everything like a warm, enzymatic ocean. And that saliva, it's not just water. It contains amylase, an enzyme that starts breaking down carbohydrates immediately. It contains lysozyme, an antimicrobial compound that literally punches holes in bacterial cell walls. If you're a gram-positive bacterium with a thick peptidoglycan layer, you might survive. If you're gram-negative with a thinner wall, you're already in trouble. The enzymes don't care about your life story, they just dissolve. But let's say you make it through the mouth. You're clinging to a piece of food, maybe tucked into a crevice that protected you from the teeth. The tongue, a massive muscular organ, pushes everything backward. You're sliding now, riding a wave of chewed food and saliva down the esophagus. It's dark, it's tight. The walls contract in rhythmic waves called peristalsis, squeezing you downward whether you like it or not. There's no swimming against this current. There's no escape hatch. You're going down. And then you arrive. The stomach. If the mouth was uncomfortable, the stomach is hell. The first thing that hits you is the smell. Or it would be if you had a nose. But you can sense the chemical composition of your environment. And right now, that environment is screaming danger. The pH here is around 2. That's approximately 1 million times more acidic than neutral water. It's comparable to battery acid, and you're swimming in it. Hydrochloric acid, that's what's trying to kill you now. The stomach's parietal cells pump out this corrosive substance specifically to break down food and murder anything living that dares to enter. Proteins unfold and denature in this acid bath. Cell membranes disintegrate. DNA strands snap apart like overcooked pasta. Most bacteria die within seconds. Their cellular machinery simply can't function at such an extreme pH. It's like trying to run a computer underwater, except the water is also on fire and made of acid. But it's not just the acid. The stomach is also secreting pepsin, a powerful enzyme that tears apart protein molecules. If you have any proteins in your cell wall or membrane, and you do because you're alive, pepsin is hunting them down and shredding them into amino acids. Meanwhile, the stomach walls are contracting, churning, mixing everything into a acidic slurry called chyme. You're being tumbled like laundry in the world's most hostile washing machine. The temperature, still 37 degrees, feels different now. In the mouth it was neutral. Here, combined with the acid and the mechanical churning, it accelerates your destruction. Chemical reactions happen faster at higher temperatures. That includes the reactions dissolving your cell wall. You're not just being attacked, you're being efficiently disassembled at the molecular level. Only the toughest bacteria survive the stomach. Species like Helicobacter pylori, which actually evolved to live here, produce an enzyme called urease that neutralizes acid in their immediate vicinity. They create tiny bubbles of survivable pH around themselves, like miniature force fields. They burrow into the mucus layer, coating the stomach wall, hiding from the worst of the acid. But you're not Helicobacter pylori. You're just a random bacterium from someone's hand or a vegetable. You don't have those adaptations. You're not equipped for this. If you're incredibly lucky, you might be protected by the food you came in with. If you're buried deep inside a piece of meat or grain, the outer layers might absorb enough acid to give you a chance. If you can produce a thick capsule around your cell wall, that might buy you a few extra seconds. Some bacteria can temporarily downregulate their metabolism, essentially playing dead and hoping the storm passes. It's not a great strategy, but it's better than nothing. The stomach doesn't hold food forever. After about 2 to 4 hours, depending on what the host ate, the pyloric sphincter at the bottom begins to open. Little by little, chyme is squirted into the small intestine. If you've survived this long, clinging to whatever shelter you could find, this is your chance. You're being pushed forward out of the acid bath into the next stage of the journey. But don't celebrate yet. You're battered, weakened, and barely functional, and ahead of you lies an entirely new set of horrors. The stomach was just the entrance exam. You passed somehow. But surviving the gut isn't about one challenge. It's about enduring all of them, one after another, with no rest and no mercy. The small intestine is waiting, and it has weapons you haven't even imagined yet. You made it out of the stomach. Congratulations, you're one of maybe a few thousand survivors out of the millions that entered with that piece of food. Your cell wall is pitted and scarred from the acid. Your membrane is barely holding together. But you're alive, and now you're tumbling into the small intestine. Surely it has to be better than the stomach, right? Wrong. The small intestine isn't interested in giving you a break. It's interested in extraction, absorption, and elimination, and you're very much in the elimination category. The first thing you notice is the change in pH. It's no longer the brutal acidity of the stomach. Here in the duodenum, the first section of the small intestine, the environment is more neutral, around 6 to 7 on the pH scale. Your cellular machinery sighs with relief. Enzymes that were denatured in the stomach start to refold. Proteins begin functioning again. For a brief, beautiful moment, you think you might actually survive this. And then the bile arrives. Bile is a yellowish-green fluid produced by your host's liver and stored in the gallbladder. When fatty food enters the small intestine, the gallbladder contracts and squirts bile into the mix. Its job is to emulsify fats, breaking them down into tiny droplets so enzymes can digest them more efficiently. But bile doesn't just attack fats. It attacks you. Bile salts are amphipathic molecules, meaning they have both water-loving and fat-loving ends. This makes them incredibly good at disrupting cell membranes, which are made of lipid bilayers. Your membrane, in other words. Imagine your cell membrane as a protective wall made of tiny oil molecules arranged in a double layer. Bile salts wedge themselves into that wall, prying the molecules apart, creating gaps and weaknesses. If enough bile salts accumulate, your membrane simply falls apart. Your insides spill out into the intestinal fluid, and you become a cloud of dissolved organic molecules. No ceremony, no last words. Just disintegration. Bile doesn't even notice. It's already moved on to the next victim. But bile isn't the only chemical weapon here. The pancreas, a gland sitting just outside the intestinal tract, is dumping enzymes into the small intestine like a factory working overtime. Proteases to break down proteins, lipases to break down fats, amylases to break down carbohydrates, nucleases to break down DNA and RNA. Every major biological molecule has an enzyme designed to tear it apart, and you, a bacterium, are made entirely of biological molecules. These enzymes don't distinguish between the protein in a piece of chicken and the protein in your cell wall. They don't care that you're alive. They're catalysts, molecular machines programmed to break bonds and split chains. If a protease encounters the peptidoglycan in your cell wall, it starts snipping. If a lipase finds the phospholipids in your membrane, it starts dissolving. You're not food, but you're being digested anyway. Collateral damage in the host's quest for nutrients. And then there's the physical environment. The small intestine isn't a gentle stream. It's a churning, contracting tunnel about 20 feet long, constantly in motion. Peristalsis, those rhythmic, muscular contractions, doesn't stop. It pushes everything forward. Food, fluid, enzymes, and you. The walls squeeze and release, creating waves that move at roughly 1 centimeter per second. That might not sound fast, but when you're a bacterium, 2 micrometers long, it's a relentless current you can't fight. The turbulence is intense. You're being tossed, spun, and slammed into other particles. Sometimes you collide with partially digested food. Sometimes you hit the intestinal wall, which is lined with tiny finger-like projections called villi. These villi increase the surface area for nutrient absorption, but they also create a complex maze-like terrain. Get caught between 2 villi during a contraction, and you might be crushed. Get swept into a crypt, the small pits at the base of the villi, and you might never get out. Worse, the upper small intestine still has traces of oxygen. Not much, but enough to be dangerous if you're an obligate anaerobe, a bacterium that can only survive in oxygen-free environments. Oxygen, for you, is poison. It generates reactive oxygen species, highly unstable molecules that tear through cellular components like microscopic grenades. Your DNA gets damaged. Your proteins get oxidized. Your lipids become rancid. Most gut bacteria evolved in the deep, oxygen-free zones of the colon. Up here, in the small intestine, oxygen is just one more way to die. But let's say you're lucky. Let's say you're a facultative anaerobe, a bacterium that can tolerate small amounts of oxygen. Or maybe you have enzymes like catalase or superoxide dismutase that neutralize reactive oxygen species before they kill you. You're still not safe, because patrolling the small intestine, embedded in the walls and floating in the fluid, are immune cells, lots of them. The small intestine contains more immune tissue than almost anywhere else in the body. Peyer's patches, clusters of lymphoid tissue, line the intestinal wall. Macrophages drift through the fluid, engulfing anything that looks foreign. Dendritic cells extend long arms between the epithelial cells, sampling the contents of the intestine, checking for invaders, and floating everywhere, coating everything, are antibodies, specifically immunoglobulin A, or IgA. IgA antibodies are Y-shaped proteins designed to bind to bacteria and viruses, marking them for destruction. If an IgA molecule recognizes a protein on your surface, it latches on. Once you're tagged, you're visible to every immune cell in the area. Macrophages will hunt you down and swallow you whole. Neutrophils will release toxic chemicals that burn through your cell wall. You become a target, and in this environment, targets don't last long. The small intestine might not have the brutal acidity of the stomach, but it has precision, it has specialized killing tools, and it has no mercy. There is one slim hope, the mucus layer. The intestinal wall is coated in a thick, gel-like mucus that protects the epithelial cells underneath from digestive enzymes and acid. If you can burrow into this mucus, you might find temporary shelter. The bile can't reach you as easily here. The enzymes are diluted. The immune cells have a harder time patrolling the deeper layers. Some bacteria have evolved to live in this mucus, attaching to the glycoproteins that form its structure, hiding in the folds and spaces between molecules. But the mucus isn't a safe house. It's constantly being renewed, shed, and replaced. The cells lining the intestine produce fresh mucus every few hours, and the old layer gets sloughed off and carried away with the chyme. If you're clinging to a section of mucus that's about to be discarded, you're going with it. There's no anchor strong enough, no grip firm enough. The small intestine is a river, and everything in it is flowing toward one destination, the colon, the final stretch. The place where, if you survive long enough to reach it, you might finally have a chance to settle down. But first, you have to endure the rest of this journey, and the worst is still ahead. You've been tumbling through the small intestine for hours now. The bile has stripped away parts of your outer membrane. Digestive enzymes have chewed holes in your cell wall. You've dodged immune cells, survived oxygen exposure, and somehow avoided being flushed out with the waste. And now, finally, you feel the environment beginning to change. The pH drops slightly, the oxygen disappears almost completely, the flow slows down. You're entering the colon, the promised land, the only place in the entire digestive system where bacteria can actually thrive. But here's the problem. You're not the first one to figure that out. The colon is already occupied, extremely occupied. Right now, in this 5-foot-long tube, there are approximately 38 trillion bacteria. That's not a metaphor. That's the actual estimate from microbiome research. 38 trillion individual bacterial cells, representing somewhere between 500 and 1,000 different species, all packed into a space about the size of a large garden hose. If you converted bacterial density into human terms, it would be like trying to find an apartment in a city where every square inch of land already has 100,000 people standing on it. Good luck. But you need to find a spot, because floating freely in the colonic fluid is a death sentence. Out here in the open, you're exposed to everything: immune cells, bacteriophages, competing bacteria, and the constant flow of waste material being pushed toward the rectum. If you can't attach to something solid, you'll be flushed out within hours. The average transit time through the colon is somewhere between 12 and 48 hours, depending on the host's diet and hydration. That's your window. Attach or be excreted. The most valuable real estate in the colon is the mucus layer. Just like in the small intestine, the walls of the colon are coated in a thick gel made of glycoproteins called mucins. But here, the mucus is even thicker, more structured, and stratified into 2 distinct layers. The inner layer, closest to the epithelial cells, is dense and largely sterile. The immune system patrols it aggressively, and it's packed with antimicrobial peptides. Very few bacteria can survive there. But the outer layer, that's where everyone wants to be. The outer mucus layer is like a bustling neighborhood. It's thick enough to provide protection from the flow of waste, but porous enough to allow nutrients to diffuse through. Glycoproteins form a 3-dimensional scaffold, creating tiny spaces and channels where bacteria can attach and grow. And here's the key. If you can wedge yourself into this matrix, anchor to the mucin molecules using specialized adhesion proteins on your surface, you might finally stop tumbling. You might finally have a home. But getting a spot isn't easy. Every attachment site is already claimed. Species like Bacteroides, Firmicutes, and Akkermansia have been here for years, possibly since the host was born. They've established themselves, built structures, and formed communities. They're not interested in sharing. Some bacteria produce sticky exopolysaccharides, slimy substances that help them cling to the mucus and to each other. Others have pili, tiny hair-like appendages that act as grappling hooks. If you don't have the right tools, you can't even compete. Let's say you do have adhesion proteins. You find a small gap in the mucus where the existing bacteria haven't completely covered every surface. You extend your pili, latch onto a mucin glycoprotein, and pull yourself in. Success. You're attached. But now comes the next challenge: staying attached. Because the mucus isn't static, it's alive, in a sense. The epithelial cells below are constantly secreting fresh mucus, pushing the older layers outward. Every few hours, the outermost mucus gets sloughed off and mixed into the fecal stream. If you're too close to the outer edge, you'll be swept away. If you're too deep, you'll suffocate under layers of new mucus or get attacked by immune cells. So you have to find the sweet spot, not too shallow, not too deep, and you have to reproduce fast enough to replace any cells that get dislodged, but not so fast that you outgrow your niche and become a target for immune surveillance. It's a delicate balance. Overgrow and the host's immune system notices, undergo, and a competitor will take your spot. This is colonization. It's not about arriving. It's about establishing, defending, and maintaining your territory in a constantly shifting landscape. Some bacteria take a different approach. Instead of embedding in the mucus, they form biofilms. A biofilm is essentially a bacterial city, a structured community of microbes encased in a self-produced matrix of sugars, proteins, and DNA. Biofilms are incredibly resilient. The outer layers of bacteria protect the inner layers from antibiotics, immune cells, and environmental stress. Nutrients flow in through channels and waste flows out. It's organized, cooperative, and extremely difficult to destroy. But forming a biofilm requires cooperation. You need other bacteria of your species, or at least compatible species, nearby. You need to communicate using chemical signals called quorum-sensing molecules. When enough bacteria gather in one spot and the concentration of these signals reaches a threshold, the group collectively switches into biofilm mode. They start producing the extracellular matrix. They change their gene expression. They commit to staying put. If you're a lone bacterium, freshly arrived and looking for a home, you're probably not forming a biofilm. You're joining one, if they'll have you. And here's where it gets even more complicated. Not all niches are equal. Some spots in the colon get more nutrients than others. Areas near the cecum, where the small intestine connects, receive a steady influx of partially digested food. Areas near the rectum are nutrient poor, mostly waste and dead cells. Some bacteria specialize in fiber degradation and thrive in high fiber zones. Others feed on mucus glycoproteins when food is scarce. A few survive on the metabolic byproducts of other bacteria, essentially eating leftovers. Your survival depends on picking the right niche for your metabolic capabilities. If you're a Bacteroides species, you're probably breaking down complex plant polysaccharides, long chains of sugars that the host can't digest on their own. If you're a Firmicute, maybe you're fermenting starches and producing short-chain fatty acids like butyrate, which the host's colon cells actually use as fuel. If you're Akkermansia muciniphila, you're literally eating the mucus itself, degrading mucin and living off the sugars embedded in its structure. Every species has a job, every niche has an occupant, and if you can't find your place, you don't belong. The colonization game isn't just about space, it's about timing, tools, and strategy. Early colonizers have an advantage. They claim the best spots, establish dominance, and make it harder for latecomers. But the ecosystem is always shifting. A change in the host's diet can wipe out entire populations and create opportunities for newcomers. An antibiotic can level the playing field entirely. Nothing is permanent. Not your home, not your neighbors, not even your survival. You've found a spot in the mucus. You've anchored yourself, but now you have to defend it, because in the colon, real estate is war. You've claimed your spot in the mucus. You're attached, stable, and starting to reproduce. For the first time since you entered this body, you can actually look around and take stock of your surroundings, and what you see is overwhelming. Bacteria everywhere. Trillions of them, packed so densely that if you could see them all at once, the colon would look less like a tube and more like a living, writhing carpet of microbial life. These aren't just random bacteria, they're your neighbors, your competitors, your ecosystem. And whether you survive the next few hours depends entirely on how well you can coexist with them. Let's start with the numbers, because they're staggering. The human colon contains roughly 38 trillion bacterial cells. To put that in perspective, the entire human body contains only about 30 trillion human cells. You are outnumbered in your own gut. These bacteria aren't spread evenly either. In the small intestine, there might be 10,000 to 100,000 bacteria per milliliter of fluid. But here in the colon, the concentration jumps to 100 billion to 1 trillion per milliliter. It's one of the most densely populated microbial habitats on Earth, rivaling deep-sea thermal vents and rich soil ecosystems. And it's not just one species. Current research suggests there are anywhere from 500 to over 1,000 different bacterial species living in a single human gut, though any individual probably hosts around 100 to 200 species in significant numbers. Each species has its own niche, its own preferred food source, its own survival strategy. Some are transient, just passing through. Others are permanent residents, established lineages that have lived in human guts for millennia. You, the newcomer, are surrounded by dynasties. The two most dominant groups are Bacteroidetes and Firmicutes. These two phyla make up roughly 90% of the gut microbiome in most healthy humans. Think of them as the major political parties of the gut. Bacteroidetes are the specialists. They're incredibly good at breaking down complex carbohydrates, plant fibers that the host can't digest alone. They have massive genomes packed with enzymes for degrading polysaccharides. When you eat vegetables, whole grains, or anything fibrous, Bacteroidetes go to work, chopping those long sugar chains into smaller pieces. Firmicutes, on the other hand, are the generalists. They're more diverse, more adaptable, and they thrive on a wider range of food sources. Some ferment starches, others break down proteins. Many produce butyrate, a short-chain fatty acid that the host's colon cells use as their primary energy source. In a very real sense, Firmicutes are feeding the gut itself. The host needs them, but they need the host's food. It's a partnership, symbiotic and ancient. Then there are the smaller players, numerically minor but functionally critical. Actinobacteria, which includes species like bifidobacterium, are early colonizers in infant guts and help with immune system development. Proteobacteria, a diverse group that includes some beneficial species and some opportunistic pathogens like E. coli, exist in low numbers in a healthy gut but can bloom during inflammation or illness. Verrucomicrobia, a phylum that includes Akkermansia mucinifera, specializes in degrading mucus and has been linked to metabolic health and gut barrier integrity. Each of these groups occupies a different layer, a different region, a different functional role. Some bacteria live in the mucus, others float freely in the lumen, the open space in the center of the colon. Some attach directly to the epithelial cells, forming close relationships with the host's tissue. A few even invade the crypts, the small pits in the intestinal lining, where they're sheltered from the flow but under intense immune surveillance. The colon isn't one habitat. It's a mosaic of microhabitats, each with its own microbial community. And within these communities, there's hierarchy. Some bacteria are keystone species, meaning their presence supports the survival of many others. If they disappear, the whole ecosystem can collapse. Certain Bacteroides species, for example, break down complex fibers into simpler sugars that other bacteria can then use. They're the primary degraders, the first step in a metabolic chain. Without them, secondary fermenters starve. The community fractures. Opportunistic pathogens, normally kept in check by competition, suddenly have room to grow. Other bacteria are mutualists. They don't compete, they cooperate. Some species produce vitamins like B12 or vitamin K that the host can't make on its own. In exchange, the host provides a warm, stable environment and a steady stream of food. It's a trade. The bacteria get shelter. The host gets nutrients. Both benefit. But this cooperation isn't altruistic. It's transactional. If the relationship stops benefiting one party, it ends. Then there are the opportunistic pathogens, bacteria that are technically part of the normal microbiome, but could turn dangerous under the right or wrong circumstances. Species like Clostridium difficile or certain strains of E. coli live quietly in small numbers, kept in check by competition and immune surveillance. But if the ecosystem is disrupted, say by antibiotics that wipe out the beneficial bacteria, these opportunists explode in number. They produce toxins, damage the intestinal lining, and cause disease. They were always there. They were just waiting for their chance. You, as a newcomer, have to figure out where you fit in this hierarchy. Are you beneficial, neutral, or pathogenic? The host's immune system is constantly asking that question. It's sampling bacteria, testing their surface proteins, checking their metabolic byproducts. If you produce something useful, like short-chain fatty acids or anti-inflammatory compounds, you might be tolerated, even encouraged. If you produce toxins or inflammatory signals, you'll be targeted for destruction. The immune system isn't evil. It's just protective. And it's learned, over millions of years of evolution, to distinguish helpful bacteria from harmful ones. Mostly. But here's the fascinating part. The bacteria aren't passive. They talk to each other. They use chemical signals called quorum-sensing molecules to communicate. When the concentration of these molecules reaches a certain threshold, bacteria know they're surrounded by their own kind. They change their behavior. They might start forming biofilms, producing different enzymes, or even becoming more virulent. It's coordinated, it's strategic, and it happens completely outside the host's awareness. You're living in a city that's constantly negotiating, competing, and cooperating, all at the molecular level. Some bacteria even exchange genes. Through a process called horizontal gene transfer, a bacterium can pass a piece of DNA to a completely unrelated species. Maybe it's a gene for antibiotic resistance. Maybe it's an enzyme for digesting a new type of fiber. Whatever it is, it spreads through the population, like information through a network. Evolution in the gut doesn't just happen through mutation and selection. It happens through sharing. The microbiome is a collective intelligence, adapting in real time to changes in diet, environment, and threat. Your neighbors aren't just obstacles, they're your environment. They shape the pH through their fermentation. They consume oxygen, creating the anaerobic zones you need to survive. They produce metabolites that you might feed on, or that might poison you. They occupy space, forcing you to find your niche. And they're watching, not consciously, but chemically. If you produce the wrong signal, release the wrong toxin, or grow too aggressively, they'll respond. Sometimes with competition. Sometimes with cooperation, sometimes with war. You're not alone in this gut. You never were, and you never will be. You've settled into your spot in the mucus. You've met your neighbors. You've survived the journey from mouth to colon. And now, for the first time since you arrived, you feel something primal and urgent. Hunger. You need food. Not in the way humans need food, with cravings and preferences and the option to skip a meal. You need food at the cellular level. Without it, your metabolism shuts down. Your ATP production stops. Your ribosomes halt protein synthesis. Within hours, maybe minutes, you'll starve to death. So the question isn't whether you'll eat, it's what you'll eat, where you'll find it, and who you'll have to fight to get it. The colon is not a buffet. It's a competitive marketplace where resources are limited, unpredictable, and fiercely contested. The primary food source is dietary fiber, the indigestible carbohydrates that the host's own enzymes can't break down. When your host eats vegetables, whole grains, legumes, or fruits, those complex polysaccharides travel through the stomach and small intestine largely intact. By the time they reach the colon, they're yours. Except they're not just yours. They're everyone's. And there are trillions of bacteria competing for the same molecules. Let's say the host just ate a salad. Cellulose, hemicellulose, pectin, resistant starches, all flooding into the colon in a wave of potential energy. If you're a Bacteroides species, you're equipped for this. You have a massive arsenal of carbohydrate-active enzymes, K-zymes, specialized proteins that can cleave specific bonds in specific polysaccharides. You latch onto a cellulose fiber, deploy your enzymes, and start breaking it down into glucose units that you can absorb and metabolize. You're winning. For now. But here's the problem. You're not the only one with Casamase. Other Bacteroides are doing the same thing. Firmicutes are fermenting nearby starches. Bifidobacteria are breaking down oligosaccharides. Everyone's eating, and the fiber is disappearing fast. The colon doesn't store food. What arrives gets consumed within hours. If you can't eat quickly enough, if your enzymes are slower or less efficient than your competitors, you'll be outcompeted. You'll go hungry while others thrive. Evolution in the gut isn't about the fittest in some abstract sense. It's about the fastest eater. And what happens when the host doesn't eat fiber? What if they eat a diet high in processed foods, refined sugars, and animal proteins, all of which get absorbed in the small intestine before they ever reach you? Then the colon becomes a famine zone. Suddenly, there's almost nothing to eat. The bacteria that specialize in fiber degradation, the ones that built their entire survival strategy around plant polysaccharides, start to starve. Populations crash, diversity drops, and the survivors, they start eating something else, the mucus. Mucus, the protective gel layer coating the intestinal wall, is made of glycoproteins called mucins. These are large, complex molecules with sugar chains attached. Normally, mucus is your shelter, your attachment point, your home. But when food is scarce, it becomes your emergency rations. Some bacteria, like Akkermansia muciniphila, are specialized mucus degraders. They have enzymes that can cleave the sugar chains off mucin proteins, releasing glucose, galactose, and other simple sugars. They thrive when dietary fiber is low because they're not competing for plant material. They're eating the architecture. But here's the danger. If too many bacteria start degrading mucus, the protective barrier thins. The inner mucus layer, the one that's supposed to stay sterile and keep bacteria away from the epithelial cells, starts to erode. Bacteria get closer to the gut lining The immune system detects them. Inflammation begins. What started as a survival strategy, eating mucus during a famine, becomes a threat to the entire ecosystem. The host's gut barrier weakens. Pathogens get opportunities. The whole system destabilizes, all because the host skipped their vegetable. Then there's another resource, the metabolic byproducts of other bacteria. This is where things get really interesting. Some bacteria are primary degraders. They break down complex fibers into simpler sugars, but they don't use all of those sugars. Some leak out into the environment, and other bacteria, the cross-feeders, specialize in consuming those leftovers. They can't break down cellulose themselves, but they can absorb the glucose that someone else liberated. They're scavengers, metabolically dependent on the generosity or wastefulness of others. For example, certain bacteroides break down starch and release maltose. Bifidobacteria nearby absorb that maltose and ferment it. In the process, they produce lactate. Then another group, the lactate-utilizing bacteria, consume that lactate and convert it into short-chain fatty acids like butyrate or propionate. It's a metabolic assembly line. Each species performs one step, and the byproducts fuel the next step. The whole community is interconnected through these nutrient webs. If one species disappears, the others feel it. Butyrate, by the way, is critical. It's a short-chain fatty acid produced mainly by firmicutors during the fermentation of dietary fiber. The host's colon cells use butyrate as their primary energy source. It fuels the epithelial cells, strengthens the gut barrier, and has anti-inflammatory effect. In a very real sense, your survival as a butyrate-producing bacterium directly benefits the host. You're not just living here, you're maintaining the infrastructure. And in return, the host provides you with a stable, warm environment and a steady supply of fiber, assuming they eat fiber. But not all bacteria are team players. Some are antagonistic. They produce bacteriocins, antimicrobial peptides specifically designed to kill competing bacteria. These are precision weapons. A bacteriocin might target bacteria with a specific surface receptor, punching holes in their membranes and causing them to lyse, to burst open. The attacking bacterium is immune to its own toxin, but its competitors aren't. It's chemical warfare at the microscopic scale. You're sitting in your niche, happily fermenting fiber, and suddenly a neighboring bacterium releases a toxin that shreds your cell wall. You're dead, your nutrients spill out, and the attacker absorbs them. This kind of competition is constant. Bacteria compete for attachment sites, for nutrients, for space, and for survival. Some produce acids that lower the local pH, creating an environment where only acid-tolerant species can survive. Others produce hydrogen sulfide, a toxic gas that kills oxygen-sensitive bacteria. A few even produce antibiotics, compounds that inhibit the growth of a wide range of competitors. The colon isn't a peaceful commune. It's an arms race. Every species is evolving. New weapons, new defenses, new strategies. And if you fall behind, you're eliminated. Then there's the ultimate wild card, the host's diet. Everything you eat, or rather everything the host eats, changes your world. A high-fiber diet favors fiber-degrading bacteria. A high-fat diet shifts the balance toward bile-tolerant species. A high-protein diet increases populations that ferment amino acids, often producing toxic byproducts like ammonia and hydrogen sulfide. Artificial sweeteners, which the host can't digest, pass into the colon and get fermented by bacteria. sometimes causing bloating and shifts in microbial composition. Every meal is a policy change. Every snack is a political shift, and you have no say in it. Fasting is particularly brutal. When the host doesn't eat for 12, 16, or 24 hours, the flow of nutrients into the colon slows to a trickle. Bacteria that rely on fresh dietary fiber start to starve. Populations contract. Some species switch to survival mode, down-regulating their metabolism and waiting for the next meal. Others start producing spores, dormant forms that can survive extended periods without nutrients. The colon becomes a wasteland, and when food finally arrives, it's a feeding frenzy. The survivors gorge, the population explodes, and the competition starts all over again. So yes, you're hungry, and yes, there's food, but getting it, keeping it, and converting it into energy before someone else does requires speed, specialization, and a little bit of luck. You're not just eating to live, you're eating to outcompete trillions of others who want the exact same molecules. The colon is a zero-sum game. Every calorie you consume is a calorie someone else doesn't get. And in this world, hunger isn't uncomfortable. It's fatal. You've found food. You've started to reproduce. You're growing, dividing, and claiming more space in the mucus layer. For a moment, you might actually believe you're going to make it. But here's the thing you need to understand about the gut. You're never unsupervised. Every second of every day, you're being watched. Not by eyes, but by cells. Immune cells, billions of them, embedded in the intestinal wall, floating in the tissue, patrolling the mucus. They're looking for threats, and you, a bacterium, are automatically suspicious. Welcome to life under constant surveillance. The gut contains the largest concentration of immune tissue in the entire human body. Roughly 70% of the body's immune cells reside here, in and around the intestines. That's not a coincidence. The gut is the primary interface between the internal body and the external world. Every day, the host consumes food, water, and along with it, millions of microbes, viruses, and foreign proteins. The immune system's job is to distinguish between what's beneficial, what's neutral, and what's dangerous. And it has to make that decision quickly, accurately, and without destroying the trillions of bacteria that actually help the host survive. It's a delicate balance, and mistakes are fatal, for you or for the host. Let's start with the gut-associated lymphoid tissue, or GALT. This is a network of immune structures embedded directly in the intestinal wall. It includes Peyer's patches, small clusters of lymphoid follicles that sample the contents of the gut and coordinate immune responses. It includes isolated lymphoid follicles scattered throughout the colon. And it includes millions of individual immune cells, macrophages, dendritic cells, T cells, B cells, all working together to monitor and respond to microbial activity. You're living on top of an immune fortress, and it's always armed. The first line of defense is the mucus itself. Remember that protective gel layer you're embedded in? It's not just structural, it's weaponized. The mucus contains antimicrobial peptides, small proteins that can kill bacteria by punching holes in their membranes. Defensins, cathelicidins, lysozyme, all of these are secreted by the epithelial cells and dissolve into the mucus, creating a chemical barrier. If you're a pathogenic bacterium, or if you look like one, these peptides will find you and destroy you. Your only hope is to stay in the outer mucus layer, where the concentration is lower, and to have surface modifications that make you resistant. Some beneficial bacteria have evolved tricks to avoid these antimicrobial peptides. They modify their cell wall structures to reduce binding. They produce enzymes that degrade the peptides before they can attack. They hide in biofilms where the outer layers of bacteria absorb the damage, protecting the inner layers. It's an evolutionary arms race. The host produces stronger antimicrobials. The bacteria evolve better defenses. And this has been going on for millions of years, generation after generation, in every mammalian gut on the planet. Then there are the antibodies, specifically immunoglobulin A or IgA. This is the most abundant antibody in the human body, and the gut is where most of it lives. IgA is secreted by plasma cells in the intestinal tissue and released into the mucus and the gut lumen. It's a Y-shaped protein designed to bind to bacteria, viruses, and toxins. When an IgA molecule recognizes a protein on your surface, maybe a flagellum, a pilus, or a membrane protein, it latches on, and once you're tagged, everything changes. IgA doesn't kill you directly. It's not a toxin, but it neutralizes you. It blocks your ability to attach to the intestinal wall. It clumps you together with other tagged bacteria, forming aggregates that are easier to flush out with the fecal stream. It prevents you from invading epithelial cells or crossing the gut barrier. Essentially, it marks you as something that needs to leave. And if enough IgA molecules bind to you, you will leave, whether you want to or not. You'll be swept out in the next bowel movement, excreted into the outside world. Your brief life in the colon ended by a protein you never saw coming. But how does the immune system decide which bacteria to target? It can't tag everyone. If it did, the entire microbiome would collapse, and the host would lose all the benefits of bacterial symbiosis. So, the immune system uses pattern recognition. It looks for molecular signatures associated with pathogenic bacteria. Lipopolysaccharide, or LPS, found on the outer membrane of gram-negative bacteria, is one such signature. Flagellin, the protein that makes up bacterial flagella, is another. Peptidoglycan fragments can also trigger immune responses. Here's the trick. Beneficial bacteria often modify these molecules to look less threatening. They alter their LPS to reduce its inflammatory potential. They downregulate flagellin production when they're in the gut. They communicate with the immune system using chemical signals that say, essentially, I'm not a threat, I'm helpful. And if the immune system believes them, it tolerates their presence. It doesn't attack, it watches, but it doesn't strike. This state of controlled tolerance is called immune homeostasis, and it's what allows the microbiome to exist at all. But if you slip up, if you produce the wrong signal or grow in the wrong place, tolerance ends. Dendritic cells, specialized immune cells with long, branching arms, extend between the epithelial cells and sample the contents of the gut. They're constantly tasting the environment, checking for bacterial antigens. If they detect something suspicious, they capture it, process it, and present it to T cells. The T cells then decide whether to mount an immune response. If the answer is yes, the entire immune system mobilizes. Macrophages arrive first. These are large, aggressive immune cells that engulf bacteria whole in a process called phagocytosis. They extend pseudopods, arm-like projections that wrap around you and pull you inside. Once you're inside a macrophage, you're trapped in a phagosome, a membrane-bound compartment that quickly fuses with a lysosome, an organelle filled with digestive enzymes and reactive oxygen species. The pH drops, the enzymes activate, and you're dissolved, digested alive. Your proteins, lipids, and nucleic acids are broken down into basic components and either recycled or excreted. The macrophage doesn't even slow down. It moves on to the next target. Neutrophils are even more brutal. They're short-lived immune cells that flood into areas of infection or inflammation. When they encounter bacteria, they can engulf them like macrophages, or they can release toxic chemicals directly into the environment. Reactive oxygen species, hypochlorous acid, the same chemical in bleach, and enzymes like myeloperoxidase that generate bactericidal compounds. Neutrophils essentially burn through bacterial populations. They're indiscriminate and destructive. If they're activated in your neighborhood, you're in serious danger, even if you're not the original target. And then there's the nuclear option, inflammation. If the immune system determines that the microbial balance has been disrupted, that pathogens are invading, or that the gut barrier is compromised, it triggers an inflammatory response. Cytokines, signaling proteins, flood the area. Blood vessels dilate. More immune cells are recruited. The mucus layer thickens. Antimicrobial peptide production ramps up. The entire environment becomes hostile. Even beneficial bacteria struggle to survive. Inflammation is the immune system's way of hitting the reset button. It destroys the existing microbial community and hopes that when things calm down, the right bacteria will return. But here's the paradox. The immune system needs the bacteria. Beneficial gut bacteria help train the immune system, especially during early development. They stimulate the production of regulatory T cells, immune cells that prevent overreaction and autoimmunity. They produce metabolites like short-chain fatty acids that have anti-inflammatory effects. They compete with pathogens, preventing them from establishing themselves. In a very real sense, the immune system and the microbiome are partners. But it's a partnership based on constant negotiation, surveillance, and the ever-present threat of violence. You can be the most beneficial bacterium in the gut, producing vitamins, fermenting fiber, supporting gut barrier integrity. But if you produce one wrong molecule, if you cross into the wrong tissue layer, if you grow too fast and look like an infection, the immune system will kill you. It won't hesitate, it won't apologize, it will eliminate the threat, assess the damage, and move on. Because in the gut, the immune system isn't cruel, it's protective. And protection, in this environment, requires ruthless efficiency. You're tolerated only as long as you're useful and invisible. The moment you become a problem, you become a target. The immune system is watching you from above, but there's another threat, and it's not coming from the host. It's coming from the bacteria next to you, your neighbors. The same organisms you've been living alongside, sharing space with, coexisting with in the mucus layer. Turns out, they're not all friendly. Some of them are actively trying to kill you. Welcome to chemical warfare, bacterial style, where the weapons are invisible, the battles are silent, and the casualties are absolute. Bacteria don't have claws or teeth. They can't fight physically, but they've evolved something arguably more effective: bacteriocins. These are antimicrobial peptides or proteins produced by bacteria specifically to kill other bacteria. Think of them as molecular assassins. Each bacteriocin is designed to target specific competitors, usually closely related species that occupy the same niche and compete for the same resources. If you're too similar to the bacteriocin producer, if you share the same surface receptors or membrane structures, you're vulnerable. And once a bacteriocin locks onto you, you're finished. Here's how it works. A neighboring bacterium detects that resources are running low. Maybe the host hasn't eaten enough fiber. Maybe the mucus layer is getting crowded. Whatever the reason, competition is intensifying. So the bacterium activates genes that produce a bacteriocin. The toxin is synthesized inside the cell, then secreted into the surrounding environment. It drifts through the mucus, the invisible cloud of death, until it encounters a target. That target is you. The bacteriocin binds to a receptor on your cell membrane. Different bacteriocins work in different ways, but many form pores, tiny holes that puncture your membrane. Once those pores open, your carefully controlled internal environment collapses. Ions flood in. Essential molecules leak out. Your membrane potential, the electrical charge difference that powers many cellular processes, dissipates. You can't maintain homeostasis. You can't generate energy. Your cell swells as water rushes in, and eventually, you lyse. You burst open like an overfilled balloon. Your contents spill into the mucus, and within minutes, you're nothing but debris. The bacteria that produce bacteriocins are immune to their own weapons. They have immunity proteins, specific molecules that neutralize the toxin or block it from binding. It's like wearing armor in a chemical attack. They can release the bacteriocin, kill their competitors, and absorb the nutrients released from the dying cells. It's brutal. It's efficient. And it's happening constantly, all around you. The gut isn't just a habitat. It's a battlefield where bacteria wage microscopic wars over territory and food. But bacteriocins aren't the only weapons. Some bacteria produce organic acids as byproducts of fermentation. Lactic acid, acetic acid, propionic acid. These acids lower the pH of the local environment, making it more acidic. Most bacteria have an optimal pH range. If the environment becomes too acidic, their enzymes denature, their metabolism slows, and they eventually die. Acid-producing bacteria, like certain lactobacillus species, can tolerate low pH. They've evolved proton pumps and buffering systems that protect them, but their competitors haven't. So by simply fermenting sugars and producing acid, these bacteria create a hostile environment that kills off less acid-tolerant species. It's chemical warfare disguised as metabolism. Then there's hydrogen sulfide. Some bacteria, particularly certain species in the Desulfovibrio genus, produce hydrogen sulfide gas as a byproduct of sulfate reduction. Hydrogen sulfide is toxic. At high concentrations, it inhibits cellular respiration by blocking cytochrome C oxidase, a critical enzyme in the electron transport chain. Bacteria that can't detoxify hydrogen sulfide suffocate even in an anaerobic environment. Meanwhile, the sulfide-producing bacteria thrive, having carved out a niche where few others can survive. They don't just coexist, they poison the neighborhood and move in. Some bacteria go even further. They produce antibiotics. Yes, actual antibiotics. The same kind of compounds that humans have weaponized in medicine. Bacteria have been producing these for millions of years. Streptomyces species in soil produce streptomycin. Bacillus species produce bacitracin. And even in the gut, certain bacteria produce antibiotic-like compounds that inhibit the growth of competitors. These molecules interfere with essential processes: cell wall synthesis, protein production, DNA replication. They're broad-spectrum killers, effective against a wide range of bacteria. And if you're caught in the crossfire, you don't stand a chance. But bacteria aren't just waging solo campaigns. They coordinate, they communicate. Through quorum sensing, bacteria release and detect signaling molecules called autoinducers. When the concentration of these molecules reaches a certain threshold, it indicates that a large population of the same species is present. The bacteria respond by changing their behavior. They might start producing bacteriocins en masse. They might form biofilms. They might upregulate virulence factors. It's collective action, a bacterial army mobilizing in response to a perceived threat or opportunity. And here's where it gets really interesting. Bacteria don't just compete, they also share. Through horizontal gene transfer, bacteria can pass pieces of DNA to each other, even across species. One common mechanism is conjugation, where two bacteria connect through a pilus, a thin tube, and transfer genetic material. Another is transformation, where bacteria pick up naked DNA from their environment, often from dead cells. And there's transduction, where viruses called bacteriophages accidentally carry bacterial DNA from one cell to another. Why does this matter? Because the genes being transferred often code for survival tools. Antibiotic resistance genes, toxin production genes, enzymes for degrading new food sources. If a bacterium evolves a successful defense against a competitor's bacteriocin, that gene can spread through the population in days. The gut microbiome isn't just evolving through mutation and selection, it's evolving through collaboration and information sharing. It's a genetic commons, where innovations spread rapidly, making the entire community more resilient and more competitive. But this also means that pathogenic traits can spread. If a harmless gut bacterium acquires a toxin gene from a pathogen, it can become dangerous. If antibiotic resistance genes spread through the microbiome, the beneficial bacteria become harder to distinguish from harmful ones when the host takes antibiotics. The fluidity of bacterial genetics is both a strength and a vulnerability. It allows rapid adaptation, but it also allows rapid destabilization. So you're not just fighting for survival against the immune system and the environment. You're fighting against other bacteria who have chemical weapons, metabolic strategies, and the ability to coordinate attacks. You're in an arms race where every species is evolving new offenses and defenses. Some bacteria build thicker cell walls to resist bacteriocins. Others produce enzymes that degrade incoming toxins. A few develop efflux pumps that expel harmful compounds before they can cause damage. It's constant evolution under constant pressure. And the host has no idea this is happening. From their perspective, digestion is a passive process. Food goes in, nutrients come out, waste is excreted, but inside the colon there's a molecular war. Trillions of bacteria locked in competition, cooperation, and conflict. Alliances form and break. Populations rise and fall. Entire species can be driven to extinction by a single successful competitor, and new species, arriving with the next meal, have to navigate this volatile landscape and find a way to survive. You thought the immune system was your biggest problem, but sometimes the deadliest threats come from your own kind. You've survived acid, bile, enzymes, immune cells, and bacterial warfare. But there's one more environmental factor that could kill you at any moment, and it's something most people never think about: oxygen. That invisible gas that humans need to survive is, for you, a poison. A silent, persistent, deadly poison. And the problem is, you can't escape it completely. Even here in the colon, supposedly an oxygen-free zone, there are traces of it. Enough to kill. Enough to make every breath, metaphorically speaking, a gamble. Let's start with the basics. Most gut bacteria are anaerobes. That means they live and thrive in environments without oxygen. Some are facultative anaerobes, meaning they can tolerate oxygen if they have to, but they prefer life without it. Others are obligate anaerobes, meaning oxygen is absolutely lethal. They have no defense against it. Exposure for even a few minutes can destroy their cellular machinery and kill them outright. If you're an obligate anaerobe, and statistically you probably are, oxygen is your existential threat. Why is oxygen so dangerous? Because it's reactive. Oxygen molecules love to steal electrons from other molecules, a process called oxidation. When oxygen gets inside a bacterial cell, it reacts with proteins, lipids, and DNA, creating what are called reactive oxygen species, or ROS. These include superoxide radicals, hydrogen peroxide, and hydroxyl radicals. All of them are incredibly destructive. They punch holes in membranes, denature enzymes, and break DNA strands. It's like setting off tiny grenades inside your cell, and once the damage starts, it cascades. Aerobic organisms, like humans, have evolved elaborate systems to deal with oxygen. They have enzymes like superoxide dismutase and catalase that neutralize ROS before they cause damage. They have antioxidants that absorb reactive molecules. They've built their entire metabolism around safely using oxygen to generate energy. But you, an anaerobic bacterium, don't have those tools. Or if you do, they're minimal, barely functional. You're trying to survive in an environment where even tiny amounts of oxygen can overwhelm your defenses. The colon is supposed to be anaerobic, and for the most part, it is. The lumen, the open central space, has almost no oxygen. Measurements show oxygen concentrations near zero in the deeper layers of the colon. That's because the bacteria themselves consume any available oxygen. Facultative anaerobes near the intestinal wall absorb oxygen molecules before they can diffuse deeper. It's a collective effort. The microbial community creates its own oxygen-free zone simply by existing in such high densities. Every bacterium is a tiny oxygen sink, removing molecules from the environment. But here's the problem. Oxygen is constantly leaking in. The intestinal wall is lined with blood vessels. Blood carries oxygen, and while the epithelial cells and the mucus layer act as barriers, they're not perfect. Oxygen diffuses from the blood through the tissue and into the mucus. Near the epithelial surface, oxygen concentrations can reach levels that are deadly for obligate anaerobes. There's a gradient. The closer you are to the intestinal wall, the more oxygen. The deeper into the lumen, the safer. But you can't live in the lumen. You need to attach to the mucus to avoid being flushed out, so you're stuck in a zone where oxygen is a constant, low-level threat. Some bacteria have adapted. Facultative anaerobes, like certain E. coli strains, can switch between aerobic and anaerobic metabolism. When oxygen is present, they use it to generate energy more efficiently. When it's absent, they switch to fermentation. They're flexible, opportunistic. They colonize the layer closest to the epithelium, where oxygen is highest, and act as a buffer, consuming oxygen before it reaches the obligate anaerobes deeper in the mucus. In a way, they're protecting the rest of the community, though not out of altruism. They're just exploiting a niche no one else can survive in. But if you're an obligate anaerobe, you need to stay deep. You cling to the outer mucus layer as far from the blood supply as possible. You minimize your metabolic activity to reduce oxygen damage. Some bacteria produce small amounts of enzymes like NADH oxidase, which can reduce oxygen to water without generating ROS. It's not a perfect defense, but it's something. A way to detoxify trace amounts of oxygen before they kill you. Still, it's a constant drain on your resources. Energy that could go toward growth or reproduction is instead spent on survival. And then there's inflammation. When the gut becomes inflamed, whether from infection, injury, or immune activation, blood flow to the intestinal wall increases. More blood means more oxygen. The oxygen gradient becomes steeper. Suddenly, areas that were safe are now flooded with reactive molecules. Obligate anaerobes start dying en masse. Their populations crash. The microbial community shifts. Facultative anaerobes and aerotolerant species expand into the vacated niches. The entire ecosystem restructures around the new oxygen levels. And if you're an obligate anaerobe stuck in the wrong place when inflammation hits, you're dead, no warning, no escape. Just oxidation and death. Inflammatory bowel diseases like Crohn's disease and ulcerative colitis create chronic oxygen exposure. The gut lining is constantly inflamed, constantly bleeding, constantly leaking oxygen. Studies show that in these conditions, the diversity of the microbiome drops. Obligate anaerobes, the bacteria that normally dominate a healthy colon, decline. Proteobacteria, many of which are facultative anaerobes, bloom. The balance shifts toward a less diverse, less stable community, and the host suffers. Inflammation disrupts the microbiome, and the disrupted microbiome worsens inflammation. It's a vicious cycle, and oxygen is one of the key drivers. But it's not just disease. Even normal physiological changes affect oxygen levels. After a meal, blood flow to the gut increases to support digestion and absorption. More blood means more oxygen diffusing into the tissue. For a few hours after the host eats, the oxygen gradient shifts. It's subtle, but for bacteria living on the edge of tolerance, subtle is enough. Some bacteria time their reproduction to avoid these high oxygen windows. Others have stress response systems that activate during oxygen spikes, temporarily shutting down vulnerable processes and waiting for levels to drop. Then there's the mucus turnover. Remember, the mucus layer is constantly being renewed. Fresh mucus is secreted from the base, and old mucus is sloughed off from the top. When new mucus is laid down, it's closer to the epithelial cells, closer to the blood supply, and higher in oxygen. Bacteria embedded in that fresh mucus are suddenly in a more dangerous environment. They have to adapt quickly, migrate deeper, or die. The mucus isn't just your home. It's a dynamic, shifting landscape where the rules change by the hour. Some bacteria form microcolonies, dense clusters where the outer cells shield the inner cells from oxygen. The bacteria on the surface absorb the oxygen, sacrificing themselves to protect the colony's core. It's a form of structural defense. By organizing spatially, the community creates oxygen-free pockets within an otherwise hostile environment. Biofilms work similarly. The extracellular matrix, the slimy scaffold that holds the biofilm together, slows oxygen diffusion. By the time oxygen molecules reach the deeper layers of the biofilm, they've been consumed by the outer layers. It's collaborative survival. But all of this requires energy, coordination, and luck. If oxygen levels spike too quickly, if inflammation comes without warning, if the mucus layer thins and exposes you to the blood supply, there's no defense. You can have every adaptation in the world, but oxygen is relentless. It doesn't negotiate. It doesn't tire. And it's always there, seeping in from the edges, waiting for a moment of vulnerability. You're living in a world where the air is poison, and the only reason you're still alive is because trillions of other bacteria are breathing it for you. For now, you've been living in the colon for days now, maybe weeks. You've carved out a niche in the mucus. You've navigated competition, evaded immune cells, and survived the constant low-level oxygen threat. You've even started reproducing, building a small population of genetically identical daughter cells. Things are stable. You're part of the ecosystem now. And then, without warning, everything ends. The host gets sick. And when the host suffers, you suffer 1,000 times worse. Welcome to the purge. Let's start with the most common disaster, diarrhea. The host ate something contaminated. Maybe it was undercooked chicken carrying salmonella. Maybe it was lettuce contaminated with pathogenic E. coli. Maybe it was just a norovirus from a poorly washed hand. Whatever the cause, the pathogen has triggered the host's defense systems, and the host's response is simple, brutal, and indiscriminate. Flush everything out immediately. The intestines shift into emergency mode. The epithelial cells start secreting massive amounts of water and electrolytes into the lumen. The goal is to dilute the toxins, wash out the pathogens, and expel them before they can cause more damage. Peristalsis. Those rhythmic muscular contractions accelerates. The normal, gentle waves become violent, rapid spasms. The colon, which usually takes 12 to 48 hours to move material through, now empties in a matter of hours, sometimes minutes. And you, clinging to your spot in the mucus, are caught in a flash flood. The mucus layer, your shelter, starts to dissolve. The epithelial cells ramp up mucus production, but they're also shedding the old mucus faster than ever. Entire sections of the mucus matrix break apart and get swept away. You're holding on with everything you have, your adhesion proteins straining, your pili anchored as deeply as possible. But the current is too strong. One by one, bacteria around you lose their grip and tumble into the torrent, and then it's your turn. You detach. You're free-floating now, spinning in a liquid chaos of water, mucus, undigested food, and billions of other bacteria, all being flushed toward the rectum. There's no fighting it, no swimming against the current. You're being expelled. Within hours, you'll pass through the anus and into the toilet bowl. Your life in the gut, your entire existence, ends not with violence, but with evacuation. Your waste now. Just another microbe in a diarrheal episode that the host will forget about in a day or two. For you, it's extinction. For the host, it's an immune response. That's the scale of the power imbalance. You don't matter. The system will replace you. But diarrhea, as catastrophic as it is, isn't the worst purge. That title belongs to antibiotics. Imagine this. The host develops a bacterial infection. Maybe it's strep throat. Maybe it's a urinary tract infection. Maybe it's pneumonia. The doctor prescribes antibiotics. Broad-spectrum antibiotics. The kind that kill a wide range of bacterial species. The host swallows a pill, and within hours, the antibiotic molecules enter the bloodstream, circulate through the body, and eventually make their way into the gut. And when they arrive, they don't discriminate. Antibiotics target essential bacterial processes. Some, like penicillin and amoxicillin, inhibit cell wall synthesis. Bacteria trying to grow or divide can't build proper cell walls. Their membranes rupture, they lyse. Others, like tetracycline and erythromycin, block protein synthesis by binding to ribosomes. Without functional ribosomes, Bacteria can't make the proteins they need to survive. Metabolism grinds to a halt. They starve at the molecular level. Still others, like fluoroquinolones, interfere with DNA replication. Bacteria can't reproduce. They can't repair damaged DNA. They die slowly, one broken molecule at a time. And here's the thing, the antibiotic doesn't know you're beneficial. It doesn't care that you've been producing butyrate, supporting the gut lining, competing with pathogens. All it sees is a bacterium. And bacteria are the target. Within days of antibiotic treatment, the gut microbiome collapses. Populations that took years to establish are wiped out in 72 hours. Diversity plummets. Studies show that a single course of antibiotics can reduce microbial diversity by 30 to 50%. Some species disappear entirely and never come back. Is less than #0. 7# is greater than. You, a random gut bacterium, are almost certainly killed. Maybe you're susceptible to the antibiotics mechanism. Maybe you're resistant, but the collapse of the ecosystem around you creates conditions you can't survive. Either way, your chances are grim. The few bacteria that do survive are usually the ones with antibiotic resistance genes, and once the susceptible bacteria are gone, the resistant ones bloom. They explode in population, filling the empty niches, consuming all the available resources. The post-antibiotic gut is a wasteland dominated by a few hardy survivors. It's not diverse, it's not balanced. It's barely functional. And into this wasteland, opportunistic pathogens arrive. Clostridium difficile, or C. diff, is the most notorious. Normally C. diff exists in small numbers, kept in check by competition from other bacteria. But after antibiotics wipe out the competition, C. diff has no natural enemies. It blooms, producing toxins that damage the intestinal lining, causing severe inflammation and diarrhea. The host, already weakened by the original infection and the antibiotic treatment, now faces a secondary infection that's often worse than the first. And it's a direct result of the microbiome collapse. But let's say you survive. Let's say you happen to carry a resistance gene, or you're in a deep biofilm that the antibiotic couldn't fully penetrate. You're one of the lucky few. The environment around you is barren. Most of your neighbors are dead. The mucus layer is thinning because there aren't enough bacteria producing the signals that stimulate mucus production. The immune system, suddenly exposed to fewer microbial signals, starts to lose its calibration. Inflammation increases. The gut barrier weakens. You're alive, but the world you knew is gone. Recolonization takes time. New bacteria arrive with every meal, every surface the host touches, every kiss and handshake. Some of these newcomers establish themselves, others are flushed out. Slowly, over weeks or months, the microbiome rebuilds. But it's not the same. The species composition is different. The functional balance is altered. Some metabolic pathways that existed before are lost because the bacteria that perform them are extinct. The gut is recovering, but it's not restored. It's changed, and you, if you're still around, are living in a fundamentally different ecosystem than the one you colonized. Then there are the smaller purges. Foodborne illness that causes vomiting and diarrhea but doesn't kill the host. Stress-induced changes in gut motility that speed up transit time and flush out sections of the microbiome. Even extreme dietary changes can act as a purge. A sudden shift from high fiber to zero fiber, or vice versa, starves certain populations and causes localized die-offs. The gut is constantly in flux, and while small disturbances can be absorbed, large ones reset the system entirely. The worst part? The host barely notices. They take antibiotics, feel better, and move on with their lives. They have a bout of food poisoning, recover in a few days, and forget it happened. But for the trillions of bacteria in their gut, these events are apocalyptic. Mass extinctions, ecosystem collapses, generational bottlenecks where only a tiny fraction of the population survives to rebuild. And the survivors carry the scars, genetically and ecologically, for the rest of the host's life. You're not just surviving day to day. You're surviving purge to purge, never knowing when the next one will come or if you'll make it through. The colon is your home. But it's also a war zone, where peace is temporary and disaster is always one meal, one pill, one infection away. You survived the stomach's acid bath. You remember that, right? The pH of 2, the hydrochloric acid, the feeling of your proteins denaturing and your cell wall dissolving. You thought that was the worst chemical environment you'd ever face. But here's the thing about the digestive system. It's not one environment. It's a gradient, a constantly shifting spectrum of pH levels that changes from organ to organ, region to region, even hour to hour. And as a bacterium trying to survive in the gut, you're riding a roller coaster of acidity and alkalinity that never stops moving. Let's review the journey. The mouth is roughly neutral, around pH 6.5 to 7. Saliva buffers the environment, keeping it stable enough for initial digestion and for the bacteria that live on your teeth and tongue. Then comes the esophagus, still neutral, just a passageway. And then the plunge. The stomach drops to pH 2, sometimes as low as 1.5 after a meal. It's brutal. It's designed to be. Hydrochloric acid kills most bacteria, breaks down proteins, and prepares food for the next stage. If you made it through, you're already in the top 1% of survivors. Next is the small intestine. As chyme leaves the stomach and enters the duodenum, the pH starts to rise. The pancreas secretes bicarbonate, a base that neutralizes the acid. Within the first few inches of the small intestine, the pH climbs to around 6. By the time you reach the ileum, the final section of the small intestine, it's closer to 7 neutral territory. For bacteria that survive the stomach's acid, this is relief. Enzymes start to refold, metabolic processes restart. You can breathe, metaphorically, but you're not settled yet. The small intestine isn't your destination. It's just another gauntlet. And then you enter the colon. The pH here is more complex. It varies depending on location, diet, and microbial activity. In a healthy colon, the pH ranges from about 5.5 to 7. The proximal colon, the first section, tends to be more acidic, around 5.5 to 6.5. Why? Because this is where most bacterial fermentation happens. When bacteria break down dietary fiber, they produce short-chain fatty acids: acetate, propionate, butyrate. These acids lower the pH. It's a byproduct of metabolism, but it also shapes the environment. If you're an acid-tolerant bacterium, this is perfect. You thrive in slightly acidic conditions. Your enzymes are optimized for pH 6. Your membrane transporters work best when there's a proton gradient you can exploit. But if you're acid sensitive, if your optimal pH is closer to 7, you're struggling. Your metabolism is less efficient, your growth slows, you're outcompeted by bacteria better suited to the local conditions. pH isn't just a number. It's a selection pressure. constantly filtering which species survive and which decline. As you move deeper into the colon, toward the distal regions, the pH rises. By the time you reach the rectum, it's closer to 6.5 or 7. This happens because there's less fermentation happening here. Most of the fermentable fiber has already been consumed in the proximal colon. What's left is mostly waste, dead cells, and bacteria being prepared for excretion. The environment is more neutral, less acidic. Different bacterial species dominate here. The colon isn't one habitat. It's a series of microhabitats, each with its own pH profile, its own microbial community, its own rules. But pH isn't static. It changes based on what the host eats. A high-fiber diet, rich in vegetables, whole grains, and legumes, increases fermentation. More fiber means more short-chain fatty acid production, which means a lower pH. Studies show that people who eat high-fiber diets have more acidic colons, especially in the proximal regions. This favors bacteria that are good at fermenting fiber, like Bacteroides and certain firmicutes. It's a feedback loop. The diet shapes the pH. The pH shapes the microbiome, and the microbiome shapes how the diet is processed. Conversely, a low-fiber diet, high in processed foods, refined sugars, and animal proteins, reduces fermentation. Less fiber means fewer short-chain fatty acids, which means a higher, more neutral pH. This shifts the microbial balance. Fiber-fermenting bacteria decline. Protein-fermenting bacteria, which produce different and often more toxic byproducts like ammonia and hydrogen sulfide, increase. The pH rises, the community changes, and the gut becomes less healthy. The host might not notice immediately, but over time, the effects accumulate. Inflammation increases, gut barrier function declines, disease risk rises. And then there are pH shocks. Imagine the host drinks a large glass of orange juice on an empty stomach. Citric acid floods into the system. The stomach's pH, already low, drops even further. When that acidic bolus moves into the small intestine, even with bicarbonate buffering, there's a temporary pH spike. Bacteria in the upper small intestine, which are used to a relatively stable pH of 6 to 7, suddenly face a wave of acidity. Some die. Others activate stress response genes, temporarily shutting down growth and waiting for conditions to normalize. It's a shock to the system, brief but intense. Or consider what happens when the host takes antacids or proton pump inhibitors, medications that reduce stomach acid production. The stomach's pH rises, sometimes to pH 4 or 5. This allows more bacteria to survive passage through the stomach. Suddenly, bacterial species that would normally be killed are making it into the small intestine and colon. The microbial composition shifts. Studies show that long-term use of acid-suppressing medications is associated with changes in the gut microbiome, including increased risk of infections like C. diff. By altering the pH in one part of the digestive system, the host inadvertently reshapes the entire microbial ecosystem downstream. Your survival as a bacterium depends on your ability to tolerate or adapt to these pH fluctuations. Some bacteria have sophisticated pH homeostasis systems. They have proton pumps that actively expel hydrogen ions, keeping their internal pH stable even when the external environment is acidic. They have buffering molecules that absorb excess protons. They have pH-sensing proteins that detect environmental changes and trigger adaptive responses. If you have these tools, you can survive in a wider range of conditions. If you don't, you're limited to narrow pH niches, and any deviation is dangerous. But maintaining pH homeostasis costs energy. Every proton you pump out requires ATP. Every buffering molecule you produce diverts resources from growth and reproduction. It's a trade-off. Bacteria that invest heavily in pH tolerance can survive in variable environments, but they grow slower than bacteria in stable, optimal conditions. Specialists outcompete generalists when the environment is stable, but when the environment fluctuates, generalists outlast specialists. It's a fundamental ecological principle, playing out at the microscopic scale in your gut. And pH affects more than just survival. It affects nutrient availability. Many enzymes are pH dependent. At low pH, certain bonds in polysaccharides become easier to break. At high pH, others are more accessible. The pH determines which nutrients can be efficiently extracted from the environment. It also affects the solubility of minerals and metals. Iron, essential for many bacterial enzymes, is more soluble at low pH. At high pH, it precipitates out and becomes unavailable. Bacteria have evolved iron scavenging systems, but their effectiveness depends on the local pH. Then there's the interaction between pH and other stressors. At low pH, bile salts become more toxic. At high pH, certain antimicrobial peptides are more effective. The pH modulates the severity of every other threat you face. It's not just one variable. It's a variable that amplifies or dampens all the others. A slightly acidic environment might make you more vulnerable to a competitor's bacteriocin. A neutral environment might make you more visible to immune cells. You're constantly navigating a multidimensional stress landscape where pH is one of the primary axes. The colon is a pH gradient, and you're somewhere on that gradient, adapted to a specific range, vulnerable outside of it. Every meal the host eats shifts the gradient. Every medication they take alters the baseline. And you, clinging to your spot in the mucus, have no control over any of it. You can only adapt, survive, and hope that the pH stays within your tolerance range long enough for you to reproduce. Because in the gut, the environment doesn't conform to you. You conform to it, or you disappear. You've adapted to the pH. You've survived the chemical warfare. You've evaded the immune system. But there are forces acting on you that have nothing to do with chemistry or biology. Physical forces. Mechanical stresses that crush, stretch, and tear at your fragile cell wall every single second. You're living inside a muscular tube that never stops moving, in a fluid environment where pressure and temperature are constant but unforgiving. Welcome to the physics of survival, where being in the wrong place at the wrong time means being physically destroyed. Let's start with temperature. The human body maintains a core temperature of approximately 37 degrees Celsius, which is 98.6 degrees Fahrenheit. For a bacterium, this is actually ideal. Most gut bacteria evolved to thrive at exactly this temperature. Your enzymes are optimized for it. Your membrane fluidity is calibrated to it. At 37 degrees, your metabolic reactions proceed at the perfect rate, fast enough to generate energy and grow, but not so fast that things fall apart. You're in the Goldilocks zone. But here's the catch. You have no ability to regulate your temperature. You are whatever temperature your environment is, and that environment can change. When the host develops a fever, everything shifts. A fever is the immune system's way of fighting infection. By raising the body's temperature to 38, 39, even 40 degrees Celsius, the host creates conditions that are hostile to many pathogens, but the fever doesn't discriminate. It affects all bacteria, pathogenic and beneficial alike. At 39 degrees Celsius, which is about 102 degrees Fahrenheit, your enzymes start to denature. Proteins that were perfectly folded at 37 degrees begin to unfold. Your membrane becomes too fluid, losing its structural integrity. Your metabolism speeds up, but not in a controlled way. It's chaotic, inefficient, wasteful. Some bacteria can tolerate fever temperatures. They have heat shock proteins, molecular chaperones that refold denatured proteins and prevent aggregation. They can temporarily shut down non-essential processes and redirect energy towards survival. But this costs resources. Growth stops. Reproduction halts. You're in damage control mode, just trying to make it through until the fever breaks. And if the fever lasts too long, or if it spikes too high, even heat-tolerant bacteria start to die. Their proteins denature faster than they can be repaired. Their membranes rupture. They lyse, spilling their contents into the gut, and become food for the survivors. But fever is rare. Most of the time, the temperature is stable. The real physical threat is mechanical. Peristalsis, those rhythmic muscular contractions that move food, fluid, and waste through the digestive tract. In the colon, peristalsis happens constantly. The smooth muscle layers in the intestinal wall contract in coordinated waves, squeezing the contents and pushing them toward the rectum. For the host, it's a necessary function. For you, it's a relentless physical assault. Imagine being attached to the mucus layer when a peristaltic wave passes through. The intestinal wall contracts. The lumen, the open space inside the colon, narrows dramatically. The pressure increases. The mucus layer is compressed, squeezed between the contracting muscle and the solid waste in the center. You're caught in the middle. Your adhesion proteins are straining to keep you attached. The shear forces, the physical pulling and tearing are immense. If your attachment is weak, you're ripped away and tumbled into the fecal stream. If your attachment is strong, you might hold on, but the mechanical stress can damage your cell wall, creating weak points that later rupture. Peristalsis isn't gentle. It's not a slow, rolling massage. In the colon, contractions can generate pressures of 10 to 30 millimeters of mercury. That might not sound like much, but at the microscopic scale, it's crushing. Bacteria are compressed against each other, against food particles, against the intestinal wall. Some bacteria form protective biofilms specifically to withstand this mechanical stress. The biofilm matrix, a gel-like network of sugars and proteins, absorbs and distributes the force, preventing individual cells from being crushed. But if you're not in a biofilm, if you're a lone bacterium clinging to the mucus, every contraction is a test of your structural integrity. And peristalsis isn't the only movement. There are also segmentation contractions, localized squeezes that mix the contents without moving them forward. These create turbulence, swirling flows that toss bacteria around like debris in a washing machine. You're spun, flipped, slammed into other particles. Collisions are constant. Sometimes you hit another bacterium and both of you survive. Sometimes you hit a sharp-edged fragment of undigested plant fiber and your cell wall cracks. Sometimes you're caught in a vortex and pressed against the intestinal wall with enough force to deform your membrane. The colon is not a calm, still pond. It's a chaotic, turbulent environment where physical forces dominate. Then there's osmotic pressure. The The concentration of solutes in the gut lumen varies depending on what the host eats and drinks.
When the host is well hydrated, the lumen is relatively dilute.
Water flows into the colon from the bloodstream, keeping the contents moist and the osmotic pressure low. But when the host is dehydrated, water is absorbed back into the body. The lumen becomes more concentrated. Salts, sugars, and other solutes increase in concentration, raising the osmotic pressure. For a bacterium, osmotic pressure is life or death. Your cell wall is semi-permeable. Water can flow in and out, but solutes mostly cannot. If the external environment becomes too concentrated, water is pulled out of your cell by osmosis. Your cytoplasm shrinks. Your membrane pulls away from the cell wall in a process called plasmolysis. Your metabolism slows. If dehydration continues, you enter a dormant state or die outright. Conversely, if the external environment becomes too dilute, the water rushes into your cell. You swell. And if you don't have mechanisms to expel the excess water, you burst osmotic lysis. Your membrane can't withstand the internal pressure, and you explode like an overinflated balloon. Many bacteria have osmoregulation systems. They produce or import compatible solutes, small molecules like proline, glycine, betaine, or trehalose that increase the internal osmotic pressure without disrupting cellular functions. This allows them to balance the osmotic gradient and prevent water from rushing in or out. But maintaining these systems costs energy, and in a nutrient-limited environment like the gut, energy is a precious resource. Every joule spent on osmoregulation is a joule not spent on growth or competition. Dehydration events are particularly brutal. When the host doesn't drink enough water, or when they have diarrhea and lose massive amounts of fluid, the colonic environment becomes a desert. The mucus layer thins and dries out. Bacteria embedded in the mucus are exposed to air pockets, regions where the protective gel has evaporated. Without the mucus buffer, you're vulnerable to mechanical abrasion, immune detection, and desiccation. Your cell wall, designed for a moist environment, begins to crack. Your membrane loses integrity. You're dying of thirst in a body that's 70% water, simply because none of that water is reaching you. And then there's the sheer physical transit. The average time it takes for material to move through the colon is 12 to 48 hours. That's how long you have to establish yourself, reproduce, and create a population large enough that some descendants will survive when you're inevitably flushed out. But transit time varies. A high-fiber diet speeds it up. Stress and anxiety can accelerate gut motility. Certain medications, like laxatives, dramatically increase the speed. If transit time drops to 6 or 8 hours, you don't have time to colonize. You pass through, never attaching, never reproducing, just a transient visitor in a system that's moving too fast for you to claim. Conversely, slow transit, like in cases of chronic constipation, creates different problems. Material sits in the colon for days. The water is absorbed almost completely. The feces become hard, dry, compacted. Bacteria are crushed between solid masses. The mucus layer is thin and depleted. Oxygen levels might rise because there's less microbial activity consuming it. The environment becomes stagnant, low in nutrients, high in waste products. It's not a faster death. It's a slower, grinding decline where resources dwindle and conditions deteriorate until only the hardiest, most stress-tolerant bacteria remain. You're not just facing chemical and biological threats. You're facing physics. Gravity, pressure, osmosis, temperature, mechanical force. These are the invisible hands that shape your existence. They don't care about your metabolism or your genes. They act on you simply because you're matter in a dynamic system. And unlike the immune system or competing bacteria, you can't evolve defenses against physics. You can only endure it, adapt to it, and hope your cell wall holds together long enough to pass your genes to the next generation. The colon is not just chemically hostile, it's physically brutal, and every second you survive is a small victory against forces that never rest. The mucus layer has been your home since you arrived. It's where you attached after surviving the stomach. It's where you've hidden from immune cells, sheltered from oxygen, and found your niche. The mucus is everything to you, your foundation, your shield, your food source when times are desperate. But here's what you need to understand about mucus. It was never meant to be permanent. It's not a solid structure. It's a living, flowing, constantly renewing gel that the host produces and discards on a cycle you can't control. And that cycle is about to become your biggest problem. Let's talk about what mucus actually is. It's a hydrogel, a network of large glycoproteins called mucins suspended in water. Mucins are massive molecules, some of the largest proteins the human body produces. They have a protein backbone decorated with hundreds of branching sugar chains. These sugar chains are heavily negatively charged, which makes them attract water and repel each other. When mucins are secreted from goblet cells in the intestinal lining, they expand, forming a thick, slippery gel that coats the epithelial surface. This gel is your world. The mucus layer in the colon is stratified into two distinct zones. The inner layer, closest to the epithelial cells, is dense, tightly packed, and almost completely sterile. Bacteria can't penetrate it. The mucin mesh is too tight. The antimicrobial peptides embedded in it are too concentrated. This layer exists to protect the host cells from direct bacterial contact. If bacteria breach it and touch the epithelium, the immune system responds immediately and violently. The inner mucus layer is a barrier you cannot cross. The outer layer, where you live, is looser, more porous, and colonizable. The mucin network here has larger gaps. Water and nutrients can diffuse through. Bacteria can embed themselves, attaching to the mucin glycoproteins using specialized adhesion proteins. Some bacteria have lectins, proteins that bind specifically to the sugar chains on mucins. Others have pili or fimbriae that anchor mechanically. You've likely used one of these strategies to establish yourself, but the outer mucus layer isn't stable. It's constantly being renewed, shed, and replaced. Here's how it works. Goblet cells in the epithelial lining continuously secrete fresh mucin. This new mucus is laid down at the base, pushing the older layers outward. The mucus moves like a conveyor belt, slowly but relentlessly, from the epithelium toward the lumen. The inner layer stays relatively stable because it's constantly replenished, but the outer layer, your layer, is expendable. Every few hours, the outermost mucus is sloughed off and mixed into the fecal stream. And if you're attached to that section of mucus when it gets discarded, you go with it. The turnover rate varies depending on location and health. In a healthy colon, mucus turnover happens roughly every 12 to 24 hours in the outer layer. That means you have less than a day to reproduce, establish a population, and ensure that some of your descendants migrate to fresher mucus closer to the base. If you don't, your entire lineage is flushed out. It's not malicious, it's just mechanical. The host needs to constantly renew the mucus to maintain barrier function. Your collateral damage? but mucus turnover accelerates during inflammation or infection. When the gut detects a threat, goblet cells ramp up mucus production. The mucus layer thickens. The turnover rate increases. Suddenly, instead of having 12 to 24 hours, you have 6 or 8. The conveyor belt speeds up. Bacteria that were stably colonized yesterday are being expelled today. The entire microbial community is disrupted. Some species, the ones that reproduce quickly or have strong adhesion mechanisms, can keep up. Others, the slow growers or weak attachers, are swept away. The ecosystem shifts, and you're either fast enough to stay or gone. And then there's the thickness problem. The mucus layer isn't uniformly thick. In some regions of the colon, it can be 50 to 100 micrometers deep. In others, it's thinner, maybe 20 to 30 micrometers. The thickness determines how much space is available for colonization. In thick mucus, there are more layers, more niches, more room for diverse microbial communities. In thin mucus, space is limited. And if the mucus thins too much, bacteria in the outer layer come dangerously close to the inner sterile layer, triggering immune responses. Diet affects mucus production and quality. A high-fiber diet stimulates goblet cells to produce more mucus. The short-chain fatty acids generated by bacterial fermentation, especially butyrate, directly signal the epithelial cells to increase mucin secretion. More mucus means a thicker, more protective layer. It also means more habitat for bacteria. But a low-fiber diet has the opposite effect. Mucus production drops. The layer thins. And when dietary fiber is scarce, some bacteria, desperate for nutrients, start eating the mucus itself. Mucin degradation is a double-edged sword. Bacteria like Akkermansia mucinifera specialize in breaking down mucin glycoproteins. They have enzymes that cleave the sugar chains, releasing monosaccharides like glucose, galactose, and fructose. These bacteria thrive when dietary fiber is low because they're not competing for plant polysaccharides. They're eating the architecture. And in small numbers, this is fine. Mucin degradation is part of the natural turnover process. But if too many bacteria switch to mucin degradation, the mucus layer erodes faster than it can be replaced. When the mucus layer thins excessively, the gut barrier weakens. Bacteria get closer to the epithelial cells. The immune system detects them. Inflammatory signals are released. The epithelium becomes leaky, allowing bacterial products and even whole bacteria to cross into the bloodstream. This triggers systemic inflammation, a condition linked to metabolic disorders, inflammatory bowel disease, and even neurological conditions. All because the mucus, the protective gel that separates bacteria from host tissue, broke down. And you, a bacterium feeding on mucin to survive a famine, contributed to that breakdown. But mucus isn't just habitat and food. It's also weaponized. Embedded within the mucus matrix are antimicrobial peptides, defensins, cathelicidins, lysozyme. These molecules are secreted by the epithelial cells and trapped in the mucus gel. They create a chemical gradient. The concentration is highest near the inner layer and decreases as you move outward. If you venture too deep into the mucus, toward the epithelium, you encounter higher concentrations of these peptides, and for most bacteria, that's lethal. Defensins work by inserting into bacterial membranes and forming pores. Once a pore opens, your membrane potential collapses. Ions leak out. Water rushes in. You lyse, Catholycidins have a similar mechanism but also disrupt biofilms and interfere with bacterial gene expression. Lysozyme targets peptidoglycan, the main component of bacterial cell walls, especially in gram-positive bacteria. It cleaves the bonds holding the peptidoglycan together, weakening the wall until it ruptures. The mucus isn't passive. It's actively hostile to bacteria that don't belong or that get too close. Some bacteria have evolved resistance. They modify their cell wall structures to reduce binding by antimicrobial peptides. They produce enzymes that degrade the peptides before they can attack. They form biofilms where the outer layers of bacteria absorb the damage, protecting the inner layers. But these defenses cost energy and resources. And they're not foolproof. The host is constantly evolving new antimicrobial strategies, and bacteria are constantly evolving new resistances. It's an arms race playing out in the mucus layer, invisible to the host but life or death for you. Then there's the spatial complexity. The mucus isn't a uniform gel. It has microstructures, channels, pockets, dense regions, and sparse regions. Some areas have high mucin density and low water content. Others are more fluid. The architecture creates micro-niches, tiny environments within the larger mucus layer where conditions differ. Some bacteria colonize the dense regions, where they're protected from shear forces but have limited access to nutrients. Others prefer the fluid channels, where nutrients flow more freely but mechanical displacement is a constant risk. Your survival depends on finding the right micro-niche for your physiology. And finally, there's the reality that the mucus is temporary. No matter how well you adapt, how firmly you attach, how perfectly you balance nutrient acquisition and immune evasion, the mucus you're living in will be shed. Maybe in 12 hours, maybe in 6, maybe tomorrow. You have no control over it. The host's epithelial cells will secrete fresh mucin. The old layer will be pushed outward, and you'll either reproduce fast enough to leave descendants in the newer mucus, or your entire lineage will end in a toilet bowl. The mucus is your protective blanket and your prison. It shelters you from the worst of the gut's dangers, but it also ensures that your time here is always limited. You're not building a permanent home. You're renting, and the lease is always about to expire. You've survived everything the gut has thrown at you. The acid, the bile, the immune cells, the competition, the oxygen, the pH swings, the mechanical stress, the mucus turnover. You've found a niche, you've adapted, and now, finally, you're ready to do the one thing that ensures your genetic legacy: reproduce. But here's the reality of being a bacterium in the gut. Reproduction isn't a leisurely process. It's not something you do when conditions are perfect. It's something you do constantly, desperately, under siege. Because if you stop, even for a moment, you're replaced by someone who didn't.
Bacteria reproduce through binary fission, a process where one cell divides into two genetically identical daughter cells.
In ideal laboratory conditions, with unlimited nutrients and no stressors, some bacteria can complete this process in as little as 20 minutes. 20 minutes from one cell to two. That's exponential growth. 1 becomes 2, 2 becomes 4, 4 becomes 8. Within a few hours, a single bacterium can theoretically produce millions of descendants. But the gut is not a laboratory, and conditions here are never ideal. First, you need resources. To divide, you have to replicate your entire genome, synthesize a complete set of proteins, build new membrane and cell wall material, and duplicate every essential cellular component. That requires energy, specifically ATP and building blocks, amino acids, nucleotides, lipids, sugars. In the colon, those resources are limited and contested. You're competing with trillions of other bacteria for the same molecules. If you can't acquire nutrients fast enough, your growth slows. Your division time stretches from 20 minutes to an hour to several hours or stops entirely. Let's say you've managed to gather enough resources. You begin DNA replication. Your circular chromosome, a single loop of DNA containing all your genetic information, starts to unwind. Enzymes called helicases separate the two strands. DNA polymerase moves along each strand, synthesizing a complementary copy. It's precise, efficient, and absolutely critical. Any error in replication could introduce a mutation. Most mutations are neutral or harmful. A few, very rarely, are beneficial. But right now, you just need an accurate copy. The process takes time, energy, and uninterrupted focus, metaphorically speaking. But you're not in a quiet lab, you're in the gut. While you're replicating your DNA, a peristaltic wave might compress you, physically stressing your cell wall. An immune cell might tag you with an antibody, marking you for destruction. A competitor might release a bacteriocin that starts punching holes in your membrane. Reactive oxygen species from inflammation might damage your DNA mid-replication, forcing your repair enzymes to stop and fix the damage before continuing. Reproduction in the gut is like trying to build a house during an earthquake while someone throws rocks at you. Assuming DNA replication completes successfully, you now have 2 copies of your genome. Next, you need to segregate them, pulling 1 copy to each end of the cell so that when you divide, each daughter cell gets a complete set of genetic instructions. Bacteria don't have a mitotic spindle like eukaryotic cells. Instead, they use simpler protein-based systems that attach to the DNA and the cell membrane, physically dragging the chromosomes apart. It's elegant but fragile. If the cell is damaged, if the membrane is compromised, if resources run out mid-segregation, the process fails. You might end up with daughter cells that have incomplete genomes. They're not viable. They die immediately. Now comes the actual division. A protein ring, made of a protein called FTZ, forms around the middle of the cell, like a belt tightening. This ring contracts, pinching the cell in two. New cell wall material is synthesized at the division site, creating a septum that eventually separates the 2 daughter cells. When the septum is complete, you're no longer 1 cell, you're 2. Each daughter cell is roughly half the size of the original, but they'll grow, consuming nutrients and synthesizing new material until they're ready to divide again. If everything goes perfectly, your population doubles. Your lineage continues. But division is a vulnerable moment. When the cell wall is being synthesized at the septum, there's a temporary weak point. If a bacteriocin attacks during division, if osmotic pressure spikes, if a macrophage engulfs you mid-division, the process fails catastrophically. Both daughter cells die. Your investment in replication, in protein synthesis, in energy expenditure, is lost. You've spent resources and gained nothing. In a resource-limited environment like the gut, that's a death sentence not just for you, but for your potential lineage. And here's the mathematical problem. Reproduction allows exponential growth, but only if conditions permit sustained division. In the gut, they don't. You're constantly being killed by immune cells, by competitors, by physical forces, by starvation. You're constantly being displaced by mucus turnover, by peristalsis, by diarrhea. The population growth rate, the net increase in bacterial numbers, is the balance between reproduction and death. If you can divide every hour and produce 2 cells, but one of those cells dies within the hour, your population stays flat. You're running just to stay in place. This is why bacterial populations in the gut reach a carrying capacity. There's a maximum density the environment can support, determined by nutrient availability, space, and the rate of removal. In the human colon, that density is roughly 100 billion to 1 trillion bacteria per milliliter of content. Once you reach that density, growth stops. Not because bacteria stop dividing, but because the death rate equals the birth rate. Every new cell that's born is balanced by another cell dying somewhere else in the population. It's equilibrium, brutal and precise. But equilibrium can shift. If the host eats a high-fiber meal, nutrients flood the colon. Suddenly, there's more food. Bacteria that can metabolize that fiber reproduce faster. Their populations boom, but this boom is temporary. Within hours, the fiber is consumed, the excess bacteria starve or are flushed out, and the system returns to equilibrium. These population fluctuations happen constantly, driven by the host's diet, health, and behavior. You're not building a stable population. You're riding a wave that rises and crashes repeatedly. Then there's the issue of genetic variation. Binary fission produces clones, genetically identical copies. That's efficient, but it's also risky. If the environment changes, if a new antibiotic appears, if the host's immune system develops a response that targets your species, every single one of your descendants is equally vulnerable. You all have the same weaknesses. A population of clones can be wiped out by a single selective pressure. This is where horizontal gene transfer becomes critical. Through conjugation, transformation, or transduction, bacteria can acquire new genes from other bacteria, even different species. Maybe you pick up a plasmid carrying an antibiotic resistance gene. Maybe you absorb DNA from a dead neighbor that codes for a new enzyme. These genes integrate into your genome or exist as separate genetic elements. When you divide, you pass them to your daughter cells. Suddenly, your lineage has a trait it didn't evolve over millions of years. It acquired it in hours. This genetic flexibility is one of the reasons bacteria are so successful. They don't just mutate, they share. But reproduction has costs beyond resources and vulnerability. Every time you divide, there's a chance of error. DNA polymerase is accurate, but not perfect. It makes roughly 1 mistake per billion base pairs copied. For a bacterial genome of a few million base pairs, that means a mutation every few hundred to few thousand divisions. Most mutations are silent or slightly deleterious, but occasionally one is beneficial. Maybe it makes you slightly more resistant to acid. Maybe it improves your ability to metabolize a specific sugar. That mutation spreads through the population if it provides an advantage. Evolution in action, happening in real time, in your gut. Some bacteria, when conditions become unbearable, stop reproducing altogether. They enter a dormant state or form spores, highly resistant structures that can survive extreme stress, desiccation, heat, acid, starvation. Spores don't grow, they don't metabolize. They just wait, sometimes for years, until conditions improve. Then they germinate, resume normal function, and start dividing again. It's a survival strategy for bacteria that can't compete in the active, crowded gut. They opt out, go dormant, and hope the future is kinder. But most gut bacteria don't form spores. They can't afford to wait. The gut is too dynamic. If you stop reproducing, your niche is filled by someone else within hours. So you divide as fast as your resources and environment allow. You push through the stress, the damage, the constant threat of death, because reproduction isn't optional. It's the only way to ensure that some version of you, some carrier of your genetic information, survives into the next generation. You're not trying to live forever. You're trying to make sure that when you die, you're replaced by your own descendants, not someone else's. That's the game, and the only way to win is to never stop playing. You've fought hard. You've adapted to every challenge the gut has thrown at you. You've found food, evaded immune cells, reproduced, and established a foothold in the mucus layer. You might even think you've earned your place here, that you've proven yourself worthy of survival. But here's the truth you need to understand. The truth that every bacterium in the gut eventually learns. You are not in control. You never were. The host decides your fate, and the host has no idea you even exist. Every decision the host makes, every meal they eat, every medication they take, every stressful day they endure, reshapes your world. And you have no say in it. You can't communicate with the host. You can't negotiate. You can't explain that the antibiotic they just swallowed is about to wipe out your entire species, or that the diet they started yesterday is starving you, or that the probiotic supplement they're taking contains competitors who will displace you. You're a passenger in someone else's body, and that body is piloted by a being who doesn't know you're there and wouldn't care if they did. Let's start with diet, because nothing shapes the gut microbiome more powerfully than what the host eats. When the host switches from a high-fiber diet to a low-fiber, high-fat diet, the entire ecosystem shifts within days. Fiber-fermenting bacteria like Bacteroides and Prevotella, which dominated when vegetables and whole grains were abundant, suddenly have nothing to eat. Their populations crash. Meanwhile, bile-tolerant bacteria that thrive on fats, like certain firmicutes and proteobacteria, explode in number. The community structure changes. Diversity drops. And you, if you were adapted to the old diet, are now obsolete. Studies show that extreme dietary changes can alter the gut microbiome composition by 30 to 50% in as little as 3 to 5 days. 3 days. That's how long it takes for the host to completely reshape the microbial world you've spent weeks colonizing. And the host isn't doing this maliciously. They're just trying a new diet, maybe for weight loss, maybe for health reasons, maybe just because they felt like it. They have no idea that inside their colon, entire bacterial populations are collapsing and new ones are rising to take their place. To them, it's a lifestyle change. To you, it's an apocalypse. Then there are antibiotics. We've talked about this before, but it's worth repeating because it's the single most destructive thing the host can do to the microbiome. A course of broad-spectrum antibiotics, taken for an ear infection or a sinus infection or strep throat, kills bacteria indiscriminately. The antibiotic doesn't just target the pathogen causing the infection. It kills gut bacteria, skin bacteria, oral bacteria, any bacterium that's susceptible to its mechanism. And gut bacteria, because they exist in such high densities and are constantly exposed to whatever the host ingests, take the hardest hit. One study found that a single course of antibiotics can reduce gut microbial diversity by up to 50%, and some species never recover. They're gone, extinct from that host's gut, and the species that do recover often come back in different proportions. creating a microbiome that's functionally different from the one that existed before. The host takes a pill. You disappear. It's that simple. And the host, once they feel better, moves on with their life. They don't think about the trillions of bacteria they just killed. Why would they? They were trying to survive an infection. You're just collateral damage. But diet and antibiotics aren't the only ways the host controls your fate. Stress matters too. When the host is stressed, their body releases cortisol and other stress hormones. These hormones affect gut motility, increasing or decreasing the speed at which material moves through the intestines. They also affect mucus production, immune function, and even the permeability of the gut barrier. Chronic stress can lead to a condition called leaky gut, where the tight junctions between epithelial cells loosen, allowing bacteria and bacterial products to cross into the bloodstream. This triggers systemic inflammation. The immune system, detecting bacteria where they shouldn't be, goes into overdrive. Inflammatory cytokines flood the gut. Neutrophils and macrophages attack. The microbial community is destabilized. Beneficial bacteria decline. Opportunistic pathogens, which thrive in inflammatory environments, increase. And you, a peaceful resident trying to ferment fiber and produce butyrate, are suddenly living in a war zone. All because the host had a stressful week at work or a fight with a loved one. Their emotional state became your physical reality. Then there's the gut-brain axis, a bidirectional communication network between the gut and the brain. The brain sends signals to the gut via the vagus nerve and hormones, affecting motility, secretion, and immune responses. But the gut also sends signals to the brain. Bacteria produce metabolites, neurotransmitter precursors, and signaling molecules that can influence mood, behavior, and even cognition. Short-chain fatty acids like butyrate, produced by bacterial fermentation, have anti-inflammatory effects and can cross the blood-brain barrier, affecting neural function. In other words, you're not just living in the gut, you're potentially influencing the host's brain. Studies suggest that the gut microbiome plays a role in anxiety, depression, and stress resilience. A healthy, diverse microbiome correlates with better mental health. A disrupted microbiome correlates with higher rates of mood disorders. You're a bacterium, and yet you might be affecting whether the host feels happy or sad, calm or anxious. It's a strange, almost eerie level of influence, but it's not intentional. You're just producing metabolites as part of your normal metabolism. The fact that those metabolites affect the host's brain is an accident of biochemistry. And yet, despite this influence, you're still powerless. You can't choose what metabolites to produce. You can't decide to help the host feel better. You're just metabolizing whatever nutrients are available and excreting the byproducts. The host, meanwhile, makes decisions based on their mood, their stress, their cravings, and those decisions loop back to affect you. If the host is stressed and craves junk food, they eat junk food. The microbiome shifts toward less diverse, less beneficial communities. Those communities produce different metabolites. The host's mood worsens, they crave more junk food. It's a feedback loop, and you're caught in it, unable to break the cycle or even understand it. The host's age also determines your fate. In infants, the gut microbiome is unstable, rapidly changing as the immune system develops and diet shifts from breast milk to solid food. In adults, the microbiome stabilizes, reaching a relatively consistent composition that can last for years. But in the elderly, the microbiome destabilizes again. Diversity drops, beneficial bacteria decline, potentially pathogenic bacteria increase. This shift correlates with increased inflammation, weakened immune function, and higher disease susceptibility. You're living through the host's entire life cycle, from birth to death, and each stage reshapes your environment. And then there's geographic relocation. If the host travels to a different country, they're exposed to new foods, new water sources, new bacteria. Some travelers develop traveler's diarrhea, a condition where their gut microbiome can't handle the local pathogens or unfamiliar dietary components. The microbiome crashes. New bacteria hitchhiking on foreign food colonize the gut. When the host returns home, the microbiome gradually shifts back, but it's never quite the same. You might survive the trip, you might not. Either way, the host's decision to travel, something completely unrelated to you, determines whether your lineage continues. Probiotics and prebiotics are another way the host, often unknowingly, manipulates the microbiome. Probiotics are live bacteria consumed as supplements meant to improve gut health. Prebiotics are non-digestible fibers that feed beneficial bacteria. If the host starts taking a probiotic containing lactobacillus or bifidobacterium species, billions of new competitors flood into the gut. If they take a prebiotic like inulin, they're selectively feeding bacteria that can metabolize it. You benefit if you're one of those bacteria. You suffer if you're not. The host is trying to be healthy. You're just trying not to be replaced. The ultimate irony is that you need the host to survive. But the host doesn't need you specifically. They need a functional microbiome. Yes. But they don't need your particular species. If you disappear, another bacterium will fill your niche. The host won't notice. Their digestion will continue. Their immune system will adapt. Life goes on. You're essential as a category, as part of the collective microbiome, but individually, you're replaceable, expendable, forgotten. You're a tenant in a body that doesn't acknowledge your existence. You're subject to forces beyond your comprehension, dietary trends, medical interventions, emotional states, aging, travel, stress. The host makes choices, and you live or die by them. You can adapt, you can evolve, you can reproduce as fast as possible and hope some of your descendants survive the next catastrophe. But you can't control the catastrophe itself. You're not the landlord. You're not even the property manager. You're just trying to survive in someone else's world, governed by someone else's rules, for as long as the host allows. And the host, blissfully unaware, continues living their life, never knowing the microscopic empire they carry within. You've been alive for a while now, maybe a few hours, maybe a day, maybe, if you're incredibly lucky, a few days. For a bacterium in the gut, that's a lifetime. You've seen things most bacteria never survive long enough to witness. Peristaltic waves, immune attacks, competitor assaults, dietary shifts. You've reproduced. You've passed on your genes. You've left descendants scattered through the mucus layer, each one carrying a copy of your DNA into an uncertain future. But now, your time is running out. Death is coming. And in the gut, death comes in many forms. Let's start with the most common cause, mechanical expulsion. You're attached to the outer mucus layer, and that layer is being shed. It's part of the normal turnover cycle, the host's way of maintaining a clean, functional gut barrier. Fresh mucus is secreted from below. Old mucus is pushed outward. And you, clinging to a section of mucus that's reached the end of its life cycle, are about to be discarded. You feel the attachment weakening. The mucin fibers you've been anchored to are breaking down, dissolving into the fecal stream. Your adhesion proteins lose their grip, And then, suddenly, you're free floating. You tumble through the lumen, swept along by peristalsis. There's no shelter here, no mucus to hide in, no biofilm to protect you. You're exposed to the full force of the colonic flow. Other bacteria collide with you. Undigested food particles buffet you. The current carries you inexorably toward the rectum. Within hours, you pass through the anus and into the toilet bowl. Your brief existence in the human gut ends not with violence or drama, but with a flush. Your waste now. Excrement. Your cell wall will eventually break down. Your DNA will fragment. Your proteins will denature. You'll return to the basic elements you were made from: carbon, nitrogen, oxygen, phosphorus. The circle closes. But not all deaths are so passive. Some are violent. Imagine you're targeted by a macrophage, one of the immune system's roaming executioners. It detects you, maybe because you produced the wrong surface protein, maybe because you wandered too close to the epithelial layer, maybe just because you were unlucky. The macrophage extends pseudopods, arm-like projections of its membrane, and wraps them around you. You're pulled inside, engulfed whole, in a process called phagocytosis. You're now trapped in a membrane-bound compartment called a phagosome. The phagosome fuses with a lysosome, an organelle filled with digestive enzymes and reactive oxygen species. The pH inside drops to around 4.5, acidic enough to denature your proteins. Enzymes attack your cell wall, breaking down peptidoglycan. Reactive oxygen species tear through your membrane, creating holes and disrupting the lipid bilayer. Your DNA is shredded by nucleases. Your metabolic machinery grinds to a halt. Within minutes, you're not a living cell anymore. You're a soup of organic molecules being absorbed and recycled by the macrophage. Your existence is erased, not a trace remains. Or maybe you're killed by a competitor. A neighboring bacterium releases a bacteriocin, a toxin specifically designed to target bacteria like you. The molecule drifts through the mucus and binds to a receptor on your membrane. It inserts itself, forming a pore. Ions flood through the pore. Your membrane potential, the electrical charge difference that powers many of your cellular processes, dissipates. You can't maintain homeostasis. You can't generate ATP. Your metabolism collapses. Your cell swells as water rushes in through the pore, unable to be expelled. The pressure builds, and then you lyse. You burst open, your cytoplasm spilling into the surrounding environment. Your competitor absorbs the nutrients released from your corpse. Your death is their meal. Starvation is another common end. The host hasn't eaten enough fiber. Nutrients in the colon are scarce. You've depleted your internal energy reserves. Your ATP levels drop. Without ATP, you can't synthesize proteins, can't repair damaged DNA, can't maintain your membrane. Your metabolism slows, then stops. You enter a state of dormancy, shutting down non-essential processes to conserve what little energy remains. But dormancy isn't survival. It's just a slower death. If nutrients don't arrive soon, if the host doesn't eat something you can metabolize, you'll never wake up. Your cell will degrade slowly, consumed by enzymes leaking from damaged organelles, until there's nothing left but a fragment of cell wall and scattered DNA. Oxygen exposure is lethal for obligate anaerobes. If inflammation increases blood flow to the gut, oxygen diffuses into the mucus layer. You're an anaerobe. You have no defenses. Reactive oxygen species form inside your cell, attacking lipids, proteins, and nucleic acids. Your membranes rupture. Your enzymes denature, your DNA breaks apart. You die quickly, sometimes within minutes, as oxidation destroys every vital component of your cellular machinery. There's no pain, no awareness, just biochemical destruction at the molecular level. And then there's the antibiotic death. The host takes a pill. The antibiotic molecules enter the bloodstream, circulate through the body, and eventually reach the gut. If you're susceptible, the antibiotic binds to its target. Maybe it inhibits your ribosome, stopping protein synthesis. Without new proteins, you can't replace damaged enzymes. Your metabolism fails. Or maybe it blocks cell wall synthesis. When you try to divide, you can't build a proper septum. Your daughter cells are malformed, non-viable. They die immediately. Or maybe it interferes with DNA replication. Your chromosome can't be copied. You can't reproduce. Your lineage ends with you. The antibiotic doesn't kill you instantly. It kills you slowly by making survival impossible. But here's the thing about death in the gut. It's not the end of the story. When you die, your cell wall breaks down. Your membrane disintegrates. Your internal contents, proteins, nucleic acids, metabolites spill out into the environment. And other bacteria are waiting. They absorb your amino acids and use them to build their own proteins. They scavenge your nucleotides and incorporate them into their DNA. They consume your lipids and integrate them into their membranes. You don't just die, you're recycled. Your molecular components become part of someone else's body, someone else's lineage. In a sense, you never fully disappear. You're transformed. And there's another way you persist: horizontal gene transfer. If you carried a useful gene, maybe antibiotic resistance or a novel enzyme, that gene might be passed to other bacteria before you die. Through conjugation, you might have transferred a plasmid to a neighbor. Through transformation, another bacterium might pick up fragments of your DNA from the environment after you lyse. Your genetic information, the instructions that made you who you were, survives in another organism. You die, but your innovation lives on. Evolution doesn't care about individuals. It cares about genes. And genes are information. Information can be copied, shared, preserved. Your descendants, if you reproduce successfully, are still alive, scattered through the mucus layer, embedded in biofilms floating in the lumen. They carry your DNA, your metabolic capabilities, your adaptations. They're not you, but they're versions of you, copies. And some of them will survive longer than you did. Some might even thrive, finding better niches, encountering favorable conditions. Your lineage continues, branching and diverging, adapting to new challenges. You're dead, but the information you carried forward, the genetic legacy you contributed, persists. In the evolutionary sense, that's all that matters. The gut itself is eternal. From a bacterial perspective, individual bacteria die constantly. Every second, millions die and millions are born. The population turns over, but the community remains. The ecosystem is stable not because any individual survives, but because the collective persists. New bacteria arrive with every meal. Old bacteria are expelled with every bowel movement. The microbiome is a river, constantly flowing, constantly changing, but always present. You were a single drop in that river. You joined it briefly, contributed what you could, and then moved on. The river doesn't remember you, but it wouldn't exist without drops like you. So how's your day been? You woke up in acid, you fought through bile and enzymes, You dodged immune cells and survived bacterial warfare. You navigated oxygen, pH gradients, mechanical stress, and mucus turnover. You found food, reproduced, and passed your genes forward. And now, whether through expulsion, predation, starvation, or chemical attack, you're dying. Your average lifespan was somewhere between 20 minutes and a few days, depending on the species and conditions. By human standards, that's nothing. A blink, a breath. But you weren't measuring time in human terms. You were measuring it in divisions, in nutrient acquisitions, in survival challenges overcome, and by those measures, you lived a full life. The gut microbiome is a universe unto itself. Trillions of organisms, hundreds of species, locked in competition and cooperation, constantly adapting, evolving, dying, and being replaced. And the host, the human whose body contains all of this, has no idea. They go about their day, eating, sleeping, working, completely unaware of the microscopic civilizations thriving and collapsing inside them. To them, digestion is automatic. To you, it was everything. Your entire existence played out in the shadows of their biology, invisible and essential. You wouldn't have survived as a bacterium in the gut. Almost nothing does for long. But the fact that any bacteria survive at all, that they've turned one of the most hostile environments on Earth into a thriving ecosystem, is extraordinary. You were part of that, and now, as you fade, the system continues without you, indifferent and eternal. So, how was your day as a bacterium in the gut? You survived the acid bath of the stomach, where pH levels dropped to 2 and hydrochloric acid dissolved most of your companions. You navigated the small intestine, dodging bile salts, digestive enzymes, and the first wave of immune surveillance. You found a precarious home in the colon's mucus layer, anchoring yourself among trillions of competitors, all fighting for the same nutrients, the same space, the same slim chance at survival. You faced chemical warfare from neighboring bacteria, armed with bacteriocins and toxic metabolites. You endured constant mechanical stress from peristalsis, the rhythmic contractions that compressed, tumbled, and threatened to flush you out with every wave. You adapted to oxygen gradients that could poison you, pH swings that could denature your proteins, and temperature spikes during fever that pushed your enzymes to their breaking point. You navigated a mucus layer that was both shelter and trap, constantly renewed and constantly discarded, carrying bacteria like you toward the exit whether you were ready or not. You competed for food in an environment where nutrients were scarce, contested, and unpredictable. You evaded an immune system with 70% of the body's immune cells stationed in and around the gut, all of them watching, sampling, and ready to destroy anything that looked even slightly threatening. You reproduced under siege, dividing as fast as your resources allowed, knowing that every daughter cell you produced increased the chances that some version of your genetic information would survive into the next generation. And through it all, you had no control. The host decided your fate with every meal they ate, every medication they took, every stressful day they endured. You were a passenger in someone else's body, adapting to decisions you couldn't influence, surviving conditions you didn't choose. Your entire existence, from birth to death, played out in a matter of hours or days, invisible to the host, essential to their survival, but individually expendable and easily replaced. You wouldn't have survived as a bacterium in the gut. Almost nothing does, not for long. The average bacterial lifespan in the colon ranges from 20 minutes to a few days, depending on species, location, and luck. Death comes from immune cells, competitors, starvation, oxygen exposure, mechanical expulsion, or the countless other hazards we've discussed. And yet, despite these impossible odds, the gut microbiome thrives. 38 trillion bacteria, representing hundreds of species, have turned one of the most hostile environments on Earth into a functioning, cooperative, wildly successful ecosystem. These bacteria aren't just surviving, they're essential. They break down dietary fibers that human enzymes can't touch, releasing nutrients and producing short-chain fatty acids like butyrate that feed the cells lining your colon. They synthesize vitamins, including vitamin K and several B vitamins that your body can't make on its own. They train your immune system, teaching it to distinguish between harmless microbes and dangerous pathogens. They compete with opportunistic pathogens, preventing them from establishing infections. They even produce metabolites that travel through your bloodstream and influence your brain, affecting mood, stress response, and possibly even sleep quality. That last part is worth pausing on. The gut-brain axis, the bidirectional communication network between your digestive system and your central nervous system, is one of the most fascinating areas of modern research. Your gut bacteria produce neurotransmitter precursors, including serotonin and gamma-aminobutyric acid, known as GABA. They produce short-chain fatty acids that have anti-inflammatory effects and can influence neural signaling. They interact with the vagus nerve, sending signals directly to the brain. And emerging research suggests that the composition of your gut microbiome may influence sleep quality, circadian rhythms, and overall mental health. A disrupted microbiome, one that's low in diversity or dominated by inflammatory species, has been linked to sleep disturbances, anxiety, and depression. A healthy, diverse microbiome, rich in fiber-fermenting bacteria, correlates with better sleep, lower stress, and improved emotional resilience. You're not just digesting food in your gut. You're hosting an ecosystem that may be influencing how well you sleep tonight. The bacteria struggling to survive in your colon, the ones fighting for nutrients and evading immune cells and reproducing under constant pressure, might be affecting whether you wake up feeling rested or exhausted. And here's the most humbling part. You have more bacterial cells in your body than human cells. The microbes living on your skin, in your mouth, and especially in your gut outnumber the cells that make up what you think of as you. You're not a single organism. You're an ecosystem. A walking, talking collection of trillions of life forms, most of them invisible, all of them engaged in their own struggles for survival. You carry entire civilizations inside you, and they're fighting wars, forming alliances, evolving, reproducing, and dying right now as you listen to this. So tonight, when you slip into your clean bed with indoor plumbing, antibiotics in the medicine cabinet, and a refrigerator full of food, maybe take a moment to appreciate the microscopic world inside you, the bacteria that survived the journey from your mouth to your colon, the ones clinging to mucus, fermenting fiber, producing vitamins, and training your immune system. They're not doing it for you. They're doing it for themselves, trying to survive in an environment that kills most of them. But in the process, they've become essential to your health, your digestion, your immune function, and possibly even your mood and sleep. The gut microbiome is a reminder that life, at every scale, is a struggle. Whether you're a human navigating the complexities of modern existence or a bacterium trying to survive in a hostile digestive tract, the challenges are real, relentless, and often unforgiving. But life persists. It adapts. It finds niches in the most unlikely places. It cooperates, competes, and evolves. And somehow, against all odds, it thrives. You wouldn't survive as a bacterium in your gut, but the fact that trillions of them do, that they've turned your digestive system into one of the most densely populated ecosystems on the planet, is extraordinary. They don't have brains, consciousness, or intentions. They're just following the basic rules of biology. Acquire energy, avoid death, reproduce. And yet, collectively, they've created something far greater than the sum of their parts. A functioning, essential, irreplaceable community that keeps you alive. So the next time you eat a meal, especially one rich in fiber, remember that you're not just feeding yourself, you're feeding trillions. The next time you take an antibiotic, remember that you're not just killing a pathogen. You're reshaping an entire ecosystem. The next time you feel stressed or sleep poorly, consider that the invisible world inside you might be struggling too, responding to signals you're sending without even knowing it. You're not separate from your microbiome. You're inseparable from it, partners in survival, whether you realize it or not. If you enjoyed tonight's journey into the microscopic universe inside you, take a moment to like this video and subscribe to the channel. And let me know in the comments, did this change how you think about your gut, about the food you eat, about the invisible life forms that call your body home? I'd love to hear your thoughts. Where are you watching from, and what time is it there? What's the most surprising thing you learned tonight? And as you drift off to sleep, consider this. Right now, in the warm darkness of your digestive system, trillions of bacteria are living, dying, competing, and cooperating. They're breaking down the food you ate today. They're producing molecules that might influence your dreams. They're fighting battles you'll never see in a war that's been raging since the day you were born. You're never truly alone. You're a universe, teeming with life, carrying entire worlds within you. And those worlds, invisible and essential, are the reason you're alive at all. Sleep well, and remember, you're not just you, you're we.